the speaker. In this session, we had 9 presentation, one by foreign speaker, Professor
Good afternoon, everyone. I hope you enjoyed the delicious Indian cuisine today. Today, 5th January 2023, we gathered here on the occasion of 108th Indian Science Congress. Today, third day of program. I, Akanksha Kantiwar, welcome you all on the behalf of Department of Botany and Molecular Biology and Genetic Engineering. Let's proceed towards our program. Now I request Professor Ahmad Daphni Sir, Institute of Evolution, University of Haifa, Israel, and our today's chairperson, J.A. Tidke, ex-Pro Vice Chancellor, Amravati University, occupy seat on dais. Thank you so much, sir. Now I request Professor Daphni Sir to please occupy the dais. He's here already. Okay. Thank you so much, sir. Now I request Professor Alka Chaturvedi ma'am to welcome our today's speaker, Professor Ahmad Daphni Sir. To welcome Dr. Chaturvedi sir. Thank you so much, sir. Now I request our today's chairperson to please introduce our today's guest.
Good afternoon to one and all. Honorable Professor Ahmad Daphni on the dais, Professor Sunil Chaturvedi, and all the dignitaries present here in this August hall. The responsibility to introduce today's giant guest is on my shoulder. Why I am saying a giant? There are very few persons in world who are associated with pollination ecology. Otherwise, in our context, we have pollination independent and ecology independent. But it is not like that. And why I have been said to introduce the guest, because I was associated with him for my postdoctoral research at Haifa University, working on international project funded by European countries. So I could get the chance to associate it with such a person. He is a great scientist. Apart from that, he is a good, very good human being. While coming to Nagpur, when Professor Chaturvedi invited him, he looked that Nagpur is near to Amravati, and immediately he gave the consent to come for Indian Science Congress. This was the reason. And when I was working with him. He was continuously busy in work. I have the opportunity to go through not less than 20,000 reprints in his laboratory. How much reprint should have, the professor should have, that indicates. In our country, being a professor of botany, I should tell myself how many reprints I have. Now it's all online and everything is available. Now there is no need to have such type of reprints. And from morning 8, he used to come to laboratory, make the tea for myself and for him, and then start the work with some music, of course Israeli music, and up to 6 or more than 6 p.m. he was continuously working on the table. Used to ask some questions or discussions with the postdocs. He had published more than 200 papers. He says only 150 or 130 like that, which are published in reputed journals. Otherwise, he has the publication in Israeli journals and in local Hebrew language. He is also interested in sacred groves, and he told me that one secret thing yesterday only, that he is writing the poems in Hebrew language. 5 books he has on his record. So he is not doing only pollination ecology, and his laboratory is so small, but the work is so large. This is the feast for Indian botanist, how to undertake the work and what depth we should have for understanding the nature. He is also always associated with nature, flowers and pollen grains. He don't know administration, he don't know politics, nothing, like in our country. He knows only pollination ecology. So I extend my sincere thanks to the organizer for giving me the chance to chair the talk of this person, and it is the figure in my cap. And to be associated with Amat's Daphne, there is no good things than that. I welcome you, Professor Daphne, for your lecture, and take your leave. Thank you very much.
I brief because also I have problems with my voice. I hate microphones, so I speak slowly and clearly. I don't like this instrument. If there will be any problem, I will go to the microphone. Oh, perfect. So, this is my 5th visit in India. As you can see, I was just looking for excuse to come again. And when Dr. Chaturvedi sent me the invitation, after 5 minutes I wrote him, okay, at first I'm coming, what is the business about? And then I opened the map and I realized that it's near Amravati. I visited Amravati 18 years ago, and it's a real pleasure. So I want at first to thank you very much for your kind invitation, and also Professor Tilke for the hospitality. You are really very, very generous. We are in this visit here for 3 days. We are ready to stay here half a year. It's so beautiful and so nice. So I ask you, if you have any questions during my lecture, please interrupt me. Don't be too polite. I know that my accent is different from your accent, and I will try to go slowly. Something not clear, just stop me. It's okay. So my subject now today is the implication of deceptive pollination on taxonomy and nature conservation policy. My partners are Nicolas Veriken, now he's a professor in Belgium. He's a specialist in bee and also in agricultural pollination. And Stella Watts was a postdoc, a very hard field worker, and I don't have to tell you that today you can't make any interdisciplinary work without partners.
Okay, next one, please. See. At first, we have to see what is deception. It's a situation that we have advertisement in flowers, but we don't have reward. Most of the flowers offer the pollinators pollen or nectar in paying for the services of pollination. It's give and take. Deception is a case that we have flower with all the advertisement, color, shape, odor, but no reward at all. So most of the deceptive flowers are orchids, and Professor Chaturvedi can tell you quite a lot about it. Next. So now let's see what are the main, what we call pollination syndromes in orchids. For example, Orchis coriophora today is an Anacamptis coriophora. It has nectar, and a lot of nectar. So bees are going from one flower to the other with a lot of self-pollination. One of the ideas is that deception was developed to reduce self-pollination, but this is one of the ideas. If you are taking another orchid, Anacamptis morio, he resembled to any general flower, not to a specific flower, no reward, just advertisement. And it's quite clear that the bee is coming to look for reward, no reward, the bee is disappointed and going to another plant. So less self-pollination. Serapias, it's orchid flower that is like a hole, and solitary bees, males, are sleeping in the flowers. It's a kind of lodge. It's a little bit heated. They are coming to sleep at night. The pollination is done when they are looking in the evening in which flower to sleep. So they are going to one flower to other. During the day, they are very active in other places. Ophrys is the very famous pseudocopulation. The flower is resembled to a bee, produce hormones like female bee of specific species. So the male is going out, the male emerges before the female, he is smelling in the air a smell of female. He is coming close to the flower, it's like a female. He is trying to copulate with it, and then he realizes, sorry, it's not the real thing, and he is trying another flower. So that is pseudo-copulation. Next.
Next.
No, there's supposed to be some pictures here.
Yeah. Just click again.
Okay, in the third one. Okay. So if we are speaking about sexually deceptive orchids, the reproduction barrier is based on specificity of odor. In general, each species has specific odor, and we'll see later, and is attracting one species. But if there is any mistake of the pollinator, no genetic barriers. That means that if we have species A and species B, and will be hybridization, if by chance there is a cell pollination that will find this hybrid, we'll have a new species. In one generation, in one step. But we later on will see what is going on. But when we are speaking about food deceptive, again, there is no barriers in the pollination. Bees are going from one species to the other, and there are some post-pollination genetic barriers, but again, a lot of hybrids. So if the weak reproductive isolation mechanisms are very low, there is a lot of hybridization, a lot of introgression. That means back hybridization, and you can imagine what does it mean from taxonomic viewpoint. Next picture. Here you can see species Ophrys. If you ask me how many names are in the literature for Ophrys, there are 500 names of species. But how many real species there are? We know that in many cases there are synonyms. That depends. According to one of the authorities, there is only 17 species. Here, 253. There are some taxonomies here. And you know, in general, when you're doing this taxonomy, the debate is if you have 12 species or 15, you have 25 or 30, but not such a scale between 70 to 253, or if you are speaking about subspecies 144. Serapies is a small genus, minimum 318, Orchis between 21 to 37. That means that deception coupled with less of genetic barriers is a hell for taxonomists. So when you are giving a name of Ophrys or Orchis, you have to tell whom you are follow. For example, I will later on will speak a little bit about Ophrys fusca. You have Ophrys fusca in the meaning of Delforge so-and-so. Because if you say just Ophrys fusca, you will see it could be 44 different names. So what to choose? That means that deception in taxonomy are not good friends. Yes, please. Now, in the Mediterranean, we have a lot of endangered species. Now, according to one of the authorities, there are 83 endangered species of Ophrys. How I can know that we are speaking about the same species? You have to say, oh, 83 species according to Mr. Baumann, but it could be 20 according to somebody else. Orchis, 15 species are threatened, Serapies 14, most of them, and those are other related genera. And here we can see another problems, the clash between taxonomy and conservation. Next, please. So now see, every one of us know that the most important issues in conservation are at first endemic species and rare species. But if a given name, a given species has 42 synonyms, what to do? For example, this is Ophrys fusca. According to the monography on Ophrys, has 42 synonyms. In Israel, we call Ophrys fusca Ophrys israelitica, and it's endemic, not rare, but endemic. So if it's a species that is around the Mediterranean, including 42 names, no reason to make any measures of conservation. If it's really endemic, we have to keep it. And we know, of course, the discussion between lumpers and splitters. In orchids, most of you can see, some of the people are lumpers, are putting 42 names under one umbrella, but the problem is that most of the taxonomy that was done in orchids were done by amateurs. We call it holiday taxonomy. People from Germany, from France, came to the islands, found something new, immediately a new species. And even today with the molecular species, we have problems. There is especially the genus Orchis was splitted on molecular basis, beautiful dendrogram. You can't see any ecological reason, any justification from pollination viewpoint or biogeography, at least about this genus. So what to do? And now, if we are speaking about the regulations of the International Union for Conservation of Nature, the categories become totally useless. Even if we have what we call red book, how to decide what is rare, what is common. So they are totally impractical tool in the case of some of the orchids. Please. Now I will give some example. Here you see pseudo-copulation. A bee is trying to copulate with the flower. To our eyes, it's not 100 resemble to a bee. Maybe the colors here. But from the viewpoint of the bee, each species of Ophrys is resemble to a spatial other species. In generally, it's one by one. Let's see. If we are speaking about Ophrys, according to Delforge, he is the king of the splitters. If we are taking these 253 species, we know the pollinators only of 154 species. If there is anybody here that was working on deception in pollination, you know that you can sit near the flower and sit and sit and sit. Nothing is coming. That is the reason why this field was neglected for so many years. Now there is another tricks how to do it, but if I will have enough time, I will speak about it later. No, sorry. On average, each species is pollinated by 1.28 species of insects, and you see, and 70% of the Ophrys species are pollinated by 1 to 4 species of pollinator. So it's not 100% species specific, but still highly specific. I can tell you just one word. When we have 2 or 3 closed Ophrys in one area, each one has different blend of odor. But one mistake could be open door for a new species. Next, please. The Ophrys philippi, it's narrow endemic of southeast France. Only 10 populations are known. Seed production is very low. In some years, we didn't find even one capsule. So, pollinators are unknown. How we can make any measures of conservation? If we have no idea who are the pollinators and what are the needs of these pollinators. What I want to stress in this lecture, it's not only the pollinators, but also the needs of the pollinator, and I will show it later. Next. Another species, only here in France, we have the same problem. Next. And here you see, this species is pollinated by Andrena hatrophiana, that is known as related to Dipsaceae. In this case, we know, if you want to conserve the Ophrys, you have to take care for species of the Dipsaceae in the same area. And this is a very important lesson. That means that not the size of the nature reserve is important, by the presence of the energy supplier of the pollinator. Next. Now, you see what we see. You see, the Ophrys species are pollinated exclusively by oleilectic bees, require the presence of their preference host in the local, but also we have to take care of the female bees. Only males are pollinating the Ophrys. They need nectar in some other plants, but they need female. Female needs pollen and nectar and nesting places. So it's become a little bit more complicated. Next. And here you can see a case that it's Serapias lingua in Sardinia. Here is very, very specific Seratina cucurbitina. And this bee is nesting only on hollow stems of feniculum or of rubus. Next step. Next. And what happened? It was a place, many people come as a botanical pilgrimage to this place. So they clean the area. Rubus is not nice, it has a lot of thorn. Feniculum is not nice, so they clear the area. What happened after 5 years? All the population of the female dropped, they have no nesting material at all. And here you can see the nesting material, the larvae, and the twigs. So from the moment that somebody realized that the nesting is in these plants, they returned the plants and the population of Serapias was recovered again. Next. Now a little bit to some of my work. Here it's a very rare case of Bethesian mimicry. You see, I published it yesterday, as you can see. This is Ophrys israelitica, endemic species to Israel. The rarest orchis species in Israel. Today we call it Anacamptis israelitica, but I don't like it. In the same area, you have Belevalia flexuosa from Liliaceae that offer nectar as well as pollen. Look, they are living the same habitat, the same flowering season, the same open flowering simultaneously. And you can see the flower, all the sepals and the labellum, and here the flowers and the stalks are the same colors. And we want to say that the dots on the orchis are imitation to the stamen of Belevalia. To make it very short, a German team came to Israel with very elaborated equipment. They took photographs that imitate the real vision of bees, and they found that bees don't differ visually between the two species. Of course, at first we found pollinators that are moving from the Belevalia to the orchis and vice versa. And again, I was sitting about 6 to 7 years one pollinator. But then when we were quite desperate, I saw a bee with pollinia of orchis in the Belevalia. So I took 20 plants of Belevalia, put them near the orchis, and in 10 minutes the mystery was solved. So now we can say that Orchis israelitica is very rare case of real Bethesian mimicry of one-to-one. But they don't have the same odor, because if deceptive flower will have specific odor, the bee will learn very quickly to avoid it and to connect the deception with odor. So we also examine the odor and the response of the bees to the odor, different odors. Next. And this is a general mimic, Orchis caspia. And this species attracting pollinators from Salvia, very common, from Ancusa, from Asphodelus, from Vicia. You see, to our eyes, these flowers are not so similar. But in the field, bees are going from one species to the other, and then by chance on the way, they are visiting one or two flowers of orchids and continue. So if you know that the stigma here is very sticky, if you will take sample of the sticky stigma here, you will find pollen grain of Salvia and all the others. Today, when I'm working on deceptive species, at first I am checking the foreign pollen on the plant, and then I'm going to look for pollinators. And it's make my life easier. So we call this system general food mimic. Next, please. And here you can see Belevalia is attracting one Bombilius and other bee. Those are specific to Orchis israelitica. But the same bee is also, the same bee is also pollinating Orchis caspia. There are also hybrids between them. And here we can see a kind of web. This plant depends on 4 species of pollinators at least. It's simplified. This one is based only on one pollinator. So if I want to keep Orchis israelitica, I have to take care of the Bellivalia and to think also what are the needs of the Eucera. But this case it's not based on sex, so it's not so complicated. Next. Now to totally different world. Iris. Those irises are something like 15 centimeter in size. We call this group Iris onchocyclus. Endemic species, has some populations in the very highly populated area in Israel near the seashore. It's in a real danger. And also we have fragmented populations. And somebody finished PhD later in my lab, and he found that in each small nature reserve, he found different pollinators and different species composition, even if the distance between the two separated nature reserves was less than 100 meters. We know today that small bees don't like to cross ways, and some of them are limited to 30 or 40 meters all their life. And if there is any barrier, especially even just tractor road, most of them are not crossing the border. And it's a threatened species. Next. So no edible reward, but males are seeking to sleep at night in the flowers. So we used to come to the flowers just before dusk, just to press the flower, and then if the bee feel uneasy, she just answer us, bzz, 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 I am here. So it's very easy to collect them, or early in the morning. And it's clear, if we found, next slide, it's pollinated, next slide please. It's pollinated only by male bees of 12 species. Most of them are Synhalonia spectabilis. So now it's more complicated. I have to find what are the males are collecting only nectars. The females are collecting nectar and pollen, but also they need nesting places. So at least 12 species are involved of males, and of course 12 species of females. So what we have done next, we construct what we call pollination web. That based of 100 of observation, it means something like that. Synhalonia spectabilis are visiting all these species. The whites here shows the frequency. So means from all the visitors of Iris atropoporea, most of them are Synhalonia spectabilis, but the nectar sources of this bee are these species. Itancusa tinctoria and Hormuzakia and the Vitia, and so all those are species that are giving nectar. And here you can see the main pollinators of Iris and the main suppliers of nectar. Totally in all this association we have, it was 33 plant species and 42 bee species and wasps. 12 were involved in the pollination of Iris. So what will be the next stage? Logically, what will be the next stage on the same line of thinking? The web of the female. Next one, please. And here you see that is more complicated. 36 species of plants are supplying pollen and nectar. 72 female species are in the area. And we see Apis mellifera was dominating in the area. And by chance, it was also visiting iris to take pollen, but but the pollination rate was quite low. But that means that if I have to keep 12 species of pollinators of iris, here some of them are the pollinators, we have to keep all these species that are supplying the nectar. And you can see that most of the bees have several sources of nectar. This, has many, it's polylactic, some of them are oligolactic. That is one aspect of this point. Now, what about the flowering season? Iris must flower in the same season of the species that supply nectar for the males and nectar and pollen for the females. Next one. Just a moment, just to show you the sources of the bee. This was the area that we were working, and here different habitats that supply the pollinators. This is what we call a very small reserve. Next. Now, what about phenology? Here you have different species, start of the flowering, end of the flowering, through 3 years. If we are looking the iris, you can see the fluctuations from year to year. That means that in this year, plants that are flowering at the end of the season will meet different other species than in the beginning of the year. So that is why in 1 year, let's say one species is the most important donator of pollen and nectar. In the other year, they could be depend on another plant. And you can see the fluctuations from year to year. Sometimes it could be in iris for about 2 to 3 weeks, and in some other species even in 1 month. Next, please. So now, if I will try to sum up this example of deceptive species, I want to come to the question, what to conserve. If we are looking from the species concept or the species approach, people are looking for endemics, narrow distribution, rare species, small population. I will give you one example. Once in Holland, near Rotterdam, they have in the middle of the city a very, very rare orchid. It was once a part of the field. So they put a fence, very beautiful, and after 10 years the species disappeared. Nobody was thinking about pollinators. And we have the habitat approach. We are looking approach an habitat that is distinctive in endemics, plants, and a lot of richness, high diversity, and if it's threatened habitat. So we are taking the whole habitat if we can, and we keep it as a habitat approach. Next. So today, especially in Israel, there is also an attitude of plant micro-reserve concept. They are speaking about area less than 20 hectares. In Hebrew, we have a title for it, pots herb, a pots reserve, because it's in the size of a pot. And they make a fence around, a wardening, you are not allowed to pick flowers, and they don't care about pollinators and other resources. They really believe from the moment that we keep the area intact, you are not allowed to pick flower, you are not allowed to go out of the path, and so on. What we want to say that all these attitudes are not enough today. The next and the last slide, please. So we suggest the integrative approach, a message to take home. to conserve the endangered species in relation to the habitat and the component, rewarding and non-rewarding species, to keep only rewarding species as the example for Rotterdam, it's not enough. Males need food, males need females and need more complicated food, pollen and nectar. So we have to know the main partners of the pollination web. Like this, the most common pollinator of the iris, the male is going to 3 plants for nectar. The female, she needs at least 7 or 8, because in some species are collecting pollen, in other nectar. So at least for one bee, you need to keep at least 8 different species. And if you have now 12 species that are involved in the pollination, you need 36 or something like that, other plants to subsidize the male and the female. We have to know what is the ecological requirement and the nest sites. Some of the bees in our region are nesting in empty shells of mollusks. And those are the first that are collected by people who are taking out, are not nice and so on. You are taking the shells, some bee will have no place for nesting. Or if there is too much trampling, the soil becomes very compact and very bees don't like it. Or these twigs of funiculum on others. So we have to know what is the requirement of the bees. Of course, it's very important to know the mycorrhiza in the soil. I never done it, but it's very important to know it. So that means that the size of the area not depends on the number of the species or the population, but area that can cover the needs of all the partners, the male, the female, the nesting. And that means a lot of work. Thank you very much.
Generally, we don't have the question answers in these sessions, but Dr. Chaturvedi told me you may have 1 or 2 questions. Otherwise, we will conclude the session.
Thank you, sir, for your lecture. My question is that climate change is happening, and in India, when we are more emphasizing the social forestry and agroforestry, more endemic trees should be included, because as you suggested, we were unaware of the web which is forming and lot number of trees will be required in future. So our plan should work according to the direction which you are showing. What are the views?
So I didn't understand the exact point.
In climate change.
Yes, yes, you are right. You are perfectly right. The point is that, at least to my knowledge, policy of conservation totally ignores climate change. And I give you an example from Europe. We know that because of the melting of the snow, many plants are climbing up, and plants in the summit are disappearing. So people are helpless. What we can do? That is a good example of climate change and disappearing of species. In other places, I don't know what we can do. You know, you can't bring snow to Switzerland, you know, to keep it. But that is very important question, because we know about changes of pollinators in the space and time, in climatic gradients that is related to climate change. But we are just scratching a little bit, you know, the subject. We have no idea. And more than this, even if we will have information, what can we do? It's very sad, but we are quite helpless. In our region, it's more complicated. Most of the bees, or let's say the life cycle of most of the bees are unknown. We even don't have the full list of our bees or any key. So I know what is the bee, but what the needs, you know, such work in one place, it was a work of one postdoc and a team of 10 people for 3 years. Yes, please.
One more question.
Oh, sorry.
Thank you for opening the new avenue of ecology in relation to pollination diversity. You have mentioned in your presentation that the flower of one of the orchid has been visited frequently by the male of the insect species. My question is that, is it the male of the one species or the different species of the insect which is visiting that flower for the collection of the pollen?
This which flower? I was speaking about.
Orchid flowers.
Which flower?
Orchid flowers have been visited by the male of the insect.
Yes. So
Is the same species, male of the same species or the different species? That is the question.
Bee species, whether the males of different bee species visit the
no,
orchid.
one species of ophrys, one species of bee. One by one, only 17% of the ophrys are pollinated more than one species. The hormones are species specific. More than this, ophrys, female insects are changing the odor after copulation. Also, the flowers are changing their odor after pollination, and males are not visiting anymore. So it's so specific.
Thank you, sir. So, on behalf of the delegates, I extend you the heartfelt this was the small piece of his work, or it was a small piece of natural history of the pollination. From last 60 years, he is working with pollinators and flower, and whatever is presented here, it is abstracted things. If you go to the literature, we can have lot of papers, research articles on the orchids, and in India we have lot of orchids. One can undertake the work, but unfortunate thing is that we are not looking for pollinators. We are talking all about the conservation strategies. Even the conservation strategies, they do not include the role of the pollinators. We can lose the plant species if we don't have the pollinators, because they are very specific in their selection. If we lost one type of bee, there is a chance of losing the plant species also.
And one
So,
for the sorry.
okay, yes.
And as talking of strength, I want to give Dr. Chaturvedi a book that I edited.
Practical part. So,
Yes. Hold it. Come, sir. Yes, sir. Thank you. Sir, one more thing.
I was working. I have the draft copy of this book with me. Before publication, it was with me. Thank you, Amats, very much. Now, point is that in our scenario, we should insist the studies on pollination ecology. That is the matter. For conservation, lot of taxonomics like Professor Yadav sir sitting here, they are looking towards the point of conservation, but by and large we are ignoring these things. It should be included in syllabus. Only embryology, only taxonomy, only pollen biology, it will not serve the purpose. So this lecture was eye-opening for the people who are working in reproductive biology. On behalf of the organizer, Amats, I extend my sincere thanks to you for being with us. Thank you very much. Thank you.
Informative. Now I request to felicitate Professor Amats Daphne sir as a token of appreciation and gratitude by offering shawl and memento.
Sir, you can.
This side. We are going to stop here the session with the remark here, that is, see you later, pollinator. Thank you very much.
Yes, please.
At stall number C41. Daphne sir, let's proceed toward Symposium 1, In situ and ex situ conservation of plant biodiversity and sustainability. To chair this session, I request Professor S.K. Chaturvedi sir to be the chairperson and Professor Rajanna sir as the recorder, and I request them to occupy the seats on the dais. Ashwajit Fuljule sir to welcome Professor S.K. Chaturvedi sir.
Honorable Professor Daphne, other senior botanists, students, ladies and gentlemen. This session will be having 7 lectures, and out of that, the first lecture will be delivered by Professor S.R. Yadav on a role of education, educational and research institute in documentation, conservation, bioprospecting and utilization of biodiversity in 21st century. About Professor Yadav, I think everybody is aware of his work. He is well-known taxonomist of our country. He has been awarded FNA, I think, before last year. He did his BSc from Science College, Karad, MSc and PhD from Bombay University, Bombay, DSc from D.Y. Patil University, Kolhapur, and I would like to mention he was the topper from his PG college. He was retired as professor from Shivaji University, Kolhapur. He is he is now working as INSA senior scientist. He guided 26 PhD student and 11 MPhil student. He published more than 300 papers. They are all peer-reviewed. He handled more than 20 projects on taxonomy, plant taxonomy, particularly angiosperm taxonomy. He described 78 new species. He is life member of 10 scientific associations, including IIAT, IBS, and ISCA, Indian Science Congress Association, and other societies. Awards, he got many awards. Best Teacher Award from Shivaji University. Then Professor Sivaraj Gold Medal. This is the important Janaki Ammal Award. This is most prestigious in our country in the field of taxonomy. So he has been awarded this. Then he is a fellow of IAAT, fellow of NASI, National Academy of Sciences, India, fellow of INSA, and then he is involved in many activities of Indian Botanical Society and Indian Science Congress. I will not take much time and invite Professor Yadav to deliver his talk.
Yeah, I know. Good evening to all of you. Just now, I must thank our chairman, Professor Chaturvedi, who happens to be my friend. Definitely friend will talk good about friend, so that is the truth. One thing I should tell, I am not going to talk any research which I have done in my life. But I would like to now, really speaking, I have come here to urge or request all botanists of our country that what we need to do. Just now, our Daphne told the complexities in conservation of biodiversity. Okay, similarly, our Dr. Rajesh Tandon, he will talk about again problems in conservation. But conservation needs to be done. This is a question. Now we are in educational institutes like colleges, universities, primary schools, secondary school, all this. And what educational institutes can do in conservation, this is what I am talking all over the country, that what is our strength as a educational institutes. Okay, doing research, publishing papers, it is one part of our academic achievements. But now, botanists have to, or biologists have to shoulder lot of responsibilities in conservation. Why we have so many responsibilities, that I will talk. Initial lecture already has explained what are the problems. Who can tell us these problems? Only biologist can tell. No forest department, no other department has any idea about biodiversity. My forest officer does not know more than 10 plants in forest, what he can conserve. Right? This is the reality, all of you know. And only biologists, botanists know what is there in nature, what needs conservation, what are the ways and means of conservation. And this knowledge is with us, then conservation, whose responsibility it is? It is our responsibility. So, you agree in the beginning with my statement, right? So, role of educational and research institute in documentation, conservation, bioprospecting and utilization of biodiversity in 21st century. Number one, documentation. All our research scholars, PhD students, MSc students document biodiversity. Am I right? Of course, botanical survey people are sitting here, but I know we train our student and they go and work for BSI. And not only BSI documents the biodiversity, our universities, colleges do maximum work in documentation of biodiversity. So first problem, which is a basic need in conservation, documentation, who does? These are educational and research institutes. Then another is bioprospecting. Our student day and night working in laboratories, bringing plants, finding out active biomolecules, all that, who does? Again, our educational institutes, remember. Okay, big research institutes they are doing very specialized work, but the beginning work, bioprospecting, in different ways, not only phytochemicals, ornamentals, wild relatives of crop plants and many things, that also our educational institutes do. Then, utilization of biodiversity. How we can utilize, how we can put our plants to use, that part is also done in colleges and universities. And finally, universities, colleges, teachers and students, it is the most important class of the country which can do really contribution for conservation. This is what I have understood. See, many times many people see new species, this is mango, Mangifera indica, finish. Every species has many implications which we are not able to convince our leaders, politicians. Each species is a unique creation and every species needs to be conserved. It is unparalleled chemist. No human beings can compete with my plants as far as production of chemicals, compounds are concerned. Every species is a witness of evolutionary history of life. Every species has phytogeographical significance, different functions it performs in ecosystem, range of tolerance and distribution, spatial relationships and phylogenetic relationships. Another thing which I want to tell you, sir, all of you, what is the real capital of human being if you ask me? It is not gold, it is not papers, it is not dollars. It is a biological material which is a real material or capital needed by human beings. for their survival and betterment of the mother earth. Who will make our leaders to understand that what is the real capital, right? So finally, every plant species, every species is a biological capital of human being. So this is what I want to tell you. And see, I work in Western Ghat. Anyway, these are all these things, panoramas. People, today I was moving in this. Every morning you start the research, evening you product. These are my funding agencies, demanding for product. Now see this, the pleasure which we derive when we go to Western Ghat or any time in rainy season. Do you pay anything? Can you calculate price of this panoramas? Price of our biodiversity? No. So why people ask us that we should come with only product? Is it the only product for which and for whom? It is for only human beings. All of us, we are working for welfare of human beings, but whether we have some time to think about species other than human beings. So this is biologist problem. Other problems I can, they don't have thinking about it. Okay, so next, see this Kas plateau, right? Can you calculate its value in terms of money? Answer is no. Then see, research, educational and research institute shape the country. Really, educational institute is a backbone of the country. Unfortunately, this is the field neglected by leaders. They don't understand. It is not only technology or development which will make country. Education is in the center and they should concentrate. Research institute, primary schools, secondary schools, universities, colleges. And I already told documentation, bioprospecting, sustainable use and conservation, our role is a primary role. In various ways, I will tell you what are the ways and what we do. Train and introduce biodiversity to students. We educate society about biodiversity. Train in identification and documentation of biodiversity. Present situation, taxonomy. From all universities, there are no taxonomies to identify plants. At least in flowering plant, there are few. But in bryophyte, pteridophyte, fungi, where are the people? Right? Because this science doesn't give product. Identification doesn't give. Okay, training and knowing biodiversity, then only we can go ahead. Create awareness about biodiversity and its importance. Who does it? Okay, creating awareness of biodiversity in society, in children, who does it? Educational institute do it. Right? Research scholars create scientific information through their research, which is utilized for application. Research projects and contributions of students and research scholars in documentation, bioprospecting, sustainable use and conservation of biodiversity is recommendable. That is what I have understood as a teacher. Creation of trained manpower. What is our role? We are teachers, most of us, and some students are there. What is our role? They ask us, what is your product? What I should give? My product, our product is trained manpower which works for the country. And that is our product. We must tell that. Right? See, strength of educational institutes. It is a data in 2017. See, how many universities are there? 54 central universities, state universities, 411, deemed university 123, private universities total 875, colleges 33,000 are there in India. And see, the student enrolled for graduation is 34 million students are enrolled for graduation every year. What is your strength? In this country, in addition, there are other universities and, IITs and so on. Now, this is a strength with educational institutes. This is the manpower. And this manpower is not paid manpower. They work for their career, and that is the great advantage for teachers and educational institutes. So, what I want to say, how many angiosperms are there in India? 21,000. How many colleges are there? 33,000. If each college take one plant for conservation, every plant species of the country can be conserved. Right? So this is what I feel, this is our strength. And why student? They are very important in this country development. Students' strength, their enthusiasm, energy and potential. See, today we are seeing students moving and their energy level. Understanding and love for nature they have. Scientific knowledge about importance of biodiversity. Strong wish to care for Mother Earth. Knowledge of plant species, their identification, propagation, multiplication. Especially propagation and multiplication, who knows? We know. We teach how to remove dormancy, how to collect seeds, how to raise seedlings. All this knowledge is with us. Up-to-date knowledge of technological innovations. Now, lens in Google, put on that plant, we can identify plants, some majority of them. So this is creation of our students, right? And innovative minds, enormous energy, enthusiasm and strong desire to work. This is our qualities of our students. And if we use this force, every plant species can be conserved in India. Okay. I have great hope and confidence that students, research scholars and teaching community are playing and will play a crucial role in creation of awareness in society, documentation, bioprospecting and conservation of biodiversity. This is what I have understood. Now this is the data, I need not to go. I work in angiosperms, but for me, probably I am more concerned with fungi now. Are there mycologists who can identify fungi in India? Sir, you tell me, all of us we are in teaching field, we don't have mycologists who can identify our smurts, ruts and so many fungi. Similar is the case with other groups, right? And people tell we teach in classroom, that is taxonomy is the mother branch of biology, the most neglected mother. This is the way we go. This is the way today, what is, if I am doing some flora of some region and documenting plant, I will not be given 1 rupee. But if it is molecular biology of one plant, DNA sequencing, 1 crore. This is the way we are working. When we can convince people what is exactly needed to do anyway. See this caste plateau, right? Just see this flower diversity. It is not just income or anything. Nature is something about that. See this diversity in Anonesi, just from Western Ghat, few plants I am showing. And this diversity, don't you think we need to, when we are spending crores of rupees on chitta, by every plant is 1 chitta, right? But we are not able to convince our leaders that every species need to be. कोई minister बोलता है चिड़िया उड़ गया तो क्या फर्क पड़ता है? That is the way they abuse biodiversity, right? I am sorry, but this is our biodiversity, and this biodiversity, who will bioprospect? Our students, our research scholars who are working day and night. Every teacher here who are professors, actually our students work and we get credit as a captain, right? So this way it goes anyway. So see this diversity I want to say from Western Ghat, little bit, and such a diversity we need to conserve, and how to conserve is the question. Now, crucial role of educational institutes in documentation as the ornamental plants or screening phytochemicals from various plants. Why the relatives? With climate change, people are after why the relatives of cultivated plants. Who will tell why the relatives? Botanist has to tell. These are the wild relatives. Where they grow? Who will tell? Botanist. So we have great responsibility of conserving bioresources. See this, wider relatives of cropland. One example I will give. This is Vigna. Vigna, 3 crops we have domesticated, and about 27, 28 species are wild species. Whether that germplasm is conserved, definitely it is conserved, and where they grow, and in future, those wild relatives are very important for improvement. Okay. Now see, we talk a lot about many groups such as orchids, such as ornamentals and so on. As a botanist, have you thought which is the most important bioresource of the country, world? It is a grass. 80% of human requirement come from grass. How many of us we touch grass? Let us see, analyze ourselves, whether we know grasses, whether where they grow, how we can use. 80% of human requirements come from grasses, and if you want to conserve biodiversity, grasslands are most important for their breeding and conservation of reptiles, birds and everything, whether we have thought about it. I am asking you a question. Okay, grasses, is there any research going? No. Okay, so anyway, because grasses is a fodder for other animals. Even if I want to conserve a tiger, I need to conserve grasslands. Then only I can conserve a tiger. That is an important thing which forest department never understood, right? So educational, all these, what educational institute do? Number one, academics. Okay, basic research, university professors, college professors, they do academics, basic research, education, then utilization, awareness, creation of conservation, all these are their functions anyway. Now see, academics. What is academics? When I discovered this species, myself and Janardhanam, Trithuria concanensis. This is a plant from India. Okay, and you know, this looks like this. This is a plant. It is 1 centimeter plant. You know, it is a basal angiosperm. Lot big story can be told. In short, I should tell you, it is an academic endeavor. No product I can give with that 1 centimeter plant. What I will give? I cannot give any product. So this is that plant. It is an academic endeavor, and it is a basal angiosperm. Any flora written in India as per APG4 has to start with this plant. It is a basal. Right? So this way it has importance. Anyway, now education. All colleges, universities, what we do? When students visit our botanical gardens, our departments, we try to explain all the plants and all seeds, whatever is there. Creating awareness in society about biodiversity is a primary function of educational institutes, remember. And awareness is the most important if we want to do something with conservation. Then see, universities, colleges have talented students and they can really document biodiversity and do it. So this is for research. All they do research, they produce products. There are many products are there, and that way in research also we are. In forest department, now see, forest department want to do conservation, but they have manpower, they have money, they have land, but they don't have knowledge. Who has to help them? Right? So we conduct, educational institutes conduct workshops for them, educate them what are the endemic plants, what are the seeds, from where to collect, how to germinate. So this work also we do for conservation. Okay. Now these are the 3-4 questions. Who will answer? Sir? What to conserve? We have to answer this question. What to conserve is only biologists have to tell. Why to conserve? Reasons. When to conserve and where to conserve. All these 4 questions are to you and me. All educational institute, all academicians, all teachers and students, they have to answer this questions. Now see, this Eriniocarpus pneumoniae is a monotypic endemic tree genus of Western Ghat. Monotypic endemic tree genus of Western Ghat. Who will conserve? Right? Another, Bactharicema serrata, Otonepalium stipulaceum, Agassimalea pauciflora. All these are monotypic genera of Western Ghats, which are very ancient species. The entire history of Western Ghat or India is embedded in their genome. Now who will conserve them? Right? Is there any organization where they have made attempt to conserve these species? Whether anybody has collected seeds, then I want to ask you, whose responsibility it is? Only botanists know it. Okay? They know where they grow, when they flower, when they fruit. Then we know when to collect seed and germinate. Is it not major responsibility of ours? Money somebody may give, but conservation we have to do anyway. So these are the species, right? I will go. Then rare endemic species. This one, see, these are two species, Delphinium and this is a Halictron. Both are endemic to Western Ghat and endangered critically. Another, this is the Garcinia indica, Garcinia talboti. This is another group. Then see this Disoxylum Gotia dora and Disoxylum malvaricum. What I want to tell in last 10-15 years, I am not a phytochemist, but about 23 alkaloids have been extracted from those two plants. That is our research scholars have done it. And they are antiviral, antifungal, and so many things are there. So these are the germplasm with us, and they are, collecting them from Western Ghat itself is a, difficult task, and we have to conserve. Now this is one ecosystem, Myristica swan. Okay, it is an ecosystem existing for more than 140 million years. 140 million years, and it is now in Maharashtra only in 2.5 acres. Whose responsibility? Who identified? Biologist. Who knows about its importance? Biologist. Who can conserve them? They know the knowledge, ecology and so on. This is a unique ecosystem and restricted to 2.5 acres in Maharashtra. Of course, in Kerala and Tamil Nadu we have, but this is the Myristica swan and two species in that. This is Myristica magnifica with this skilt roots, unique system, and this is a Gymnopranthera canary which need roots. Don't you think our children should see? So how to conserve? We need ecologists, we need reproductive biologists, we need physiologists, not only taxonomy. He can only tell them, but others have to play a major role in conservation. Right? But unfortunately everybody works with crop plants. All botanists are converted into agriculturists. They don't focus on wild plants. I am sorry to say, I am saying it with confidence that most of the botanists who are working their physiological whatever experiment on cultivated plants and not on wild plants. Our role is to bring wild plants into limelight. That is our role. When we will understand that, okay, ecology do of wild plants. Why only of other this one cultivated? This is you see Arenga whitey, okay, Beltingia codopana, which is endangered species I am showing only. See this. This is Corypha umbraculifera. One example I am telling you. This is a Corypha utans, utans. Now Corypha umbraculifera, that palm produces largest panicle in the world. We teach in classroom. Largest panicle. And it grows in Western Ghat. Have you brought in garden? Students को पढ़ाते हैं. This is largest panicle. We never try to bring in botanical gardens. Is it not our duty? Is it a difficult task? Go to Yana, collect seeds, grow in gardens, they grow happily. All palms from all other countries are there in your garden, our own palms are not there. Okay, anyway. So see this, Hypini dichotomous, another palm. Okay. These are the plants which we have grown in botanical garden of Shivaji University. See this plant, Harpulia. Okay. See the fruit, beautiful color. Can we boil prospect it? Grow as a love plant. Okay, in university campus, people will start loving it and they will grow. See this? We have to do some tactics to conserve our plants. See this poisonous plant, Holigarna, Arnatiana, Holigarna, Grahama. All these are very, poisonous plants. But you know, all poisons are best medicines. So somebody has to work on their phytochemistry. These are Hopia parviflora. These are endemic plants. Hopia ponga. This Vatica. This is also very rare, found in Kerala. All these plants we are growing in our botanical garden. I am saying that these plants are what I have shown, which are rare. These we are growing. And so I told you, no, my laboratory. Everybody tells Yadav discovered 78 species. This is not truth. Truth is, our student discovered 78 species of angiosperms new to science. So that is their contribution. And of course, I was guide. For that, you can give me credit. I don't mind. So recently, one species is described. I am telling you how stories go and should go. This is Crinum malabaricum, and you know, see this species, illegal species. And up to 2013, all botanists of our country could not locate this species, could not say it is a new species. So we discovered as a new species. Okay, and see these students. I am telling you, it is not this reason. Our students, they are ready to keep any amount of efforts. If teacher says, Guru Shishya Parampara, we are very lucky. We have committed students and they are looking at those plants, right? See this shows. Okay, now see this plant, its close relative is found in Thailand. Okay, it is from Thailand. And this is found in India. Anyway, see, it is not important. See this. What I am showing, this is one new discovery, has many implications, not one. Leaves, longest leaves in genus Crinum in the entire world. This is a discovery. Leaves grow approximately 5 centimeters per day. My physiologist should work on that. Why so efficient? Okay. Then it is restricted to 0.5 square kilometer in the entire world when we reported. Now it is in 2-3 more streams it is recorded. Is it not a plant of top priority for conservation in India? Already DBT has given 2 projects to 2 students. They are doing on conservation. Now how we play major role in conservation? See, it is related to Thyanum found in Thailand. It is a tetraploid. And last point is very important. When paper is published in current science, highest galanthamine content in the bulbs, highest galanthamine content in the bulb which is used for treating disease Alzheimer. You know, Alzheimer. And that drug is very costly, but material is very low. Now my biotechnologists should come to our help and do tissue culture and see whether we can get that galanthamine synthesis in tissue culture. Okay? So this is one thing which I wanted to tell you. And see, many times we teachers, I'm sorry, I will say something, but it is coming from my heart, from my stomach. Okay? What is the role of botanist? Taxonomist leave apart. Botanist. When we are with MSc PhD degree as a botanist, first question, what is this plant? We must be able to tell scientific name. Don't say I am a meteorologist. Don't say I am a cytologist. We are botanists. Second question, where it is found? You must know different where it is found. And third question, for what it is used. These 3 questions if you cannot answer as a botanist, really it is not good on our part. So this is what I wanted to share with you. And when we are in any college, anywhere we are working, why not to work a flora of that region? Document. Biodiversity registers only we have to pay. Our NGOs have made. Am I right? Those who don't know anything, they will take Yadav's list, his list, make a biodiversity register and give. Is it a truth? Right? So, anyway. So, from one pond, how many new species are described of this whole size? How you new species. So, anyway, so that needs critical analysis. And then bioprospecting, this Nilambari, we released as a floricultural crop just to show how we can bioprospect our own plants as ornamentals. And now there is a mission on floriculture headed by Prime Minister. So this is the one. So see this. Now, restoration of critically endangered species. Critically endangered. Some time you tell me.
We were there.
Huh?
No, time. Just you take few minutes.
Few minutes, right. So these 2 species, what I want to tell, conservation of critically endangered species, again we are deeply concerned. What are the critical species? So say for example, Hubardia, Aptadurana and the Ceropedia fantastica, which are, see this, the way we have to work. Okay. Of course that strength is with forest department, but they don't have knowledge. Again we have to do. But our student can do it, right? So see this. Now these Ceropedias are from Maharashtra. Tell me, who will conserve them? Who know them? how to propagate them. Seed, a lot of problems are in pollination and seed production. So these are Ceropedias. What I want to tell as a botanist, that this is our plant wealth and we are deeply concerned with their conservation. And our biotechnologists, our classical methods, we propagate, we make seeds, and see, student it is our great strength. Always teacher, don't feel, I have only 2 hands. You have so many hands. Okay, students into 2, that many hands you have. And you can do. So with student help, we have tried to restore about 30 species of endangered species in Western Ghats. Okay, so see our students. Right? These are the students, student track. And if every college, every university does it, I don't think we need anyone else to conserve our plants, but we have to decide and make a move. Something like a chalwal, right? So these are our students, they are doing simple experiment we did. Okay, simple experiment. In forest department, you tell me what are the seedlings available? I know. Most of them are exotic, right? Faster growing. When we will have nurseries of our indigenous plants in our country, are there nurseries of indigenous plants in our country? Answer is approximately no. Okay, so we started this experiment of collecting seeds every year. You see, per year we collect 250 species, around 200, 300 species, collect seeds. Our student, our staff member, the best gift to Yadav sir is to bring some seeds. I will be very happy, okay? So these seeds, we give it to Malis, produce thousands of seedlings, not 1, 2, and then distribute to the people. Whoever comes, we give seedlings, educational institute. If Shivaji University can do, why not all other universities? 800 universities, 33,000 colleges. Each college, if it does, we can conserve many plants. You see these palms in Shivaji University. Today there are more than 115 species of palms in Lead Botanical Garden. I purchased one seedling for 2,000 rupees. Today you come, I have 10,000 seedlings of same plant in my garden. I can give free of cost. This is what we have to do the role. Over. So just a minute. Plant diversity is our biological capital, and finally I want to tell you, sir, we are the best people because we are student of plants. Number 1, wait, 2 minutes. Plants are best. Best friends. Remember, best friends, best doctors, best artists, best designers, best
Continue.
See, sir, I have to discuss, I don't want to approach upon other stuff. 3 things I want to say. Botanists should understand. Unless my stomach is full, the entire world is useless for everyone. Am I right? Whether it is technology, whether it is going via aeroplane, I am getting aeroplane ticket. That pleasure if you want to have, your stomach has to be full. And only there is one scientist on this planet which can synthesize food not only for human beings, the entire world is plant with minimum capital. Am I right? Yes. That is our capital. Second, if I want to be healthy, with disease I cannot enjoy anything. So far, whatever diseases humans have suffered, plants have come to our health to keep our health. Yes. So my health is my plants. And third, whatever I am seeing you, enjoying you, giving you lecture, I have to be alive. And my breath is my plants. Yes. And fourth thing I need is water. Only this with 4 capitals, the entire world runs except human being. We need many other things. All animals are happy more than us. With that 4 capitals, only we are not happy. And that's why plant or biodiversity is our real capital, and we are the people who know about them, and it is our great responsibility to conserve this biodiversity. Every one of us is concerned, and when you go to your colleges, please start these activities with your students, and I'm sure we can do wonderful work without money also. Thank you very much.
Talk. Professor Yadav is known for his slides and his talk. So I thank you very much, sir, for a very nice talk. And before going further, I have an announcement that we are having parallel session for posters, and we are having judges. I will announce the name of the judges from amongst the audience. They should go and judge the posters. So the hall number 1 below, Professor Arun Arya and Professor Sonali Chaturvedi. They are the judges for that. Second hall, we are having posters, Professor Harvansh Singh from Patiala and Professor Rajesh Arora. You please go. All the 4, please go. Professor Dongarwar will guide you, and please help us. Thank you very much. Now, I'll give time for 2 questions hurriedly because we are running short of time. So please ask if...
I have no questions, but I request before you that you will distribute hard copy of this lecture.
Yes, we'll do.
My personal request.
Hard copy, I will give you soft copy.
All audience and
all couriers.
Okay, okay. Yes, please. Yes.
बेसिकली सर मैं जबलपुर का हूँ
हाँ.
और सौभाग्य से माइकोलॉजिस्ट प्रोफेसर अग्रवाल, बैक्टीरियोलॉजिस्ट प्रोफेसर आदरकर।
अरे आप क्वेश्चन करिए ना।
मैं ये कहना चाहता हूँ यादव जी से, यदि राष्ट्र पर कुछ मेहरबानी करनी है तो आज के बाद अपने लेक्चर हिंदी में देना शुरू करिए, जिससे कि राष्ट्र का विकास हो और विज्ञान का लोक व्यापीकरण हो।
Okay, note it down.
बहुत बड़ी चीज़ है।
Yes, sir. Note it down. Then thank you very much, sir. Let us give a big applause to Professor Yadav for nice talk. Now, yes.
Fuljule sir to felicitate Professor S.R. Yadav, sir.
All your talks and speaker for this session is Professor Rajesh Tandon from Botany Department, Delhi University, Behavior of Trees for their conservation. Professor Rajesh Tandon, he did his PhD in the year 1998 from Delhi University. He is a student of Professor H.Y. Mohan Ram, famous botanist of the country. He produced 15 PhD and 6 in progress. Under his guidance or supervision, 5 MPhil students completed their degree. He has written 2 books and he has published more than 60 research papers in peer-reviewed journals. Apart from this, he is a reviewer in many reputed scientific journals. Apart all of this, he is very good human being. He is very polite, well-mannered, he is simple, and he is having high quality. With these words, I request Professor Tandon to start your work.
Good afternoon, everyone. And thanks, Sunil, for very kind words. Because he's a friend, he will speak very nice of me. So I have known him for some years now. My job is made easier by the earlier speakers, Professor Daphne and Professor Yadav. And I have been working on tree species with the same perspective as specialization of Professor Daphne and Sunil Chaturvedi and many others who would be doing the same kind of work. I've been working on reproductive biology of tree species since my PhD days, and I have trained some students in the same area. So in the due course, I have been able to work on nearly, say, 20-25 species of trees. But at the same time, most of this work was done under the All India Coordinated Project which was funded by Ministry of Environment and Forest. So we have some multiple information on the similar lines. So in the next 25 minutes or 20 minutes, I'd like to convince why reproductive biology of tree species is important, and in what way it it is crucial for conservation of plant genetic resources, not only in the country but elsewhere in the world as well. So we'll begin with my lecture. At the same time, I would say that it won't be an exaggeration if I say that trees are the major forest forming, growth forms. In fact, my teacher used to say, you can have a tree without a forest, but you cannot have a forest without a tree. So what are the forests then? Forests are the site where most of the species diversity is occupied. So you need to have forests, the corridors between forests, and you need to know their biology if you want to do conservation effectively. The other species, I won't call them the forest forming species, but they are dependent on forest. Your lianas, your other shrubs, herbs, annuals, what are the species occupy different areas of the tree in a different, strata of the tree. So they are dependent on tree species. So in this context, I would say that trees are very important, growth forms to study. Okay, this is a picture which has been taken from maps of the world, and what you see here is where you have, major forest in the world. So we have around 60,000 tree species in the world, that many. And this database has come from Botanical Conservation International. And you will be surprised to know that we only know about 9.12% of them in different contexts of reproduction, or maybe their survival or some ecology, and still we have scope for so many tree species to be studied. And it's important that, like Yadav was saying, we need to expand the scope of investigation, and it cannot be done by one person, but you need to have a multidisciplinary approach, then only you can achieve it. This is the nutshell what Professor Yadav spoke about. So looking at India, the picture is not very rosy. You look at not much greenery as compared to rest of the tropics, and we have pine trees only confined to the foothills of Himalaya, but we have Western Ghats and the northeast where evergreen forests are there. But where you are sitting today, you are a part of a deciduous broadleaf forest or a mixed forest, where you have species like Butea monosperma, you have species like Bucknania, you have species like Tectona, Shoria. These are the species which occupy, make this, area. And the forest cover of India is not more than 21%. And if you look at how many tree species are there in our country, we have this number which has come from, the same, source which I have given earlier. Nearly 2,603 tree species are there. Nearly 650 are endemic and 469 are threatened. We have been targeting threatened species because it's easy to convince the funding agency that why this need to be conserved so that you can have money. But side by side, you can also start working on the associates which are occurring with threatened species in the world. So now, how many species do we, actually know the different aspect of reproduction there? Only 142 out of 2,603. Only 5% of the species have been pursued from the perspective of plant reproductive biology. And there is scope for so many of them. If you look at the tree species, where you find them the most, of course, the top one is Madhya Pradesh, and you have Arunachal, Chhattisgarh, Odisha, and then Maharashtra. So we need to target, the trained manpower in this particular area so that we can take care of the tree species to be conserved. Now, what our experience is that the reproductive success of tree species mostly relies on three aspects, and that's the three main reproductive attributes: phenology, breeding system, and pollination system. And then you have, many sub-attributes. I'm not going into the details because of the paucity of the time, but the important thing is they influence each other. For example, this is one tree species which I studied during my PhD days, and it's called Acacia senegal, or a gum arabic, not a native of India, but from Africa. And this is a journalist which is visited by a number of insect species. But the interesting thing here is, when we worked on the breeding system of this species, some individuals, there is some scope for selfing also. When you compare selfing with outcrossing, outcrossing packages better attributes of survival than selfing, isn't it? So at the same time, there is a misconception from our books that plants would be either completely or obligately self-incompatible, or they would be completely self-compatible. So, but nobody thinks about that there is a very diffuse boundary between the two, and there are large number of plants which lie in terms of the breeding system in between them. They are not a mixed mating system. So, in the same species, when we, give the generate data for different individuals in the population, you will see that they behave differently. Some are obligately outcrossing species or individuals, and some permit self-pollination to some extent, so that there is reproductive assurance and the tree species is able to survive. So there is a kind of a gradation that you see within even the self-incompatible species, and acacias are generally known to be self-incompatible in rest of the world. This is another species which I worked, and it's a very abundant tree species from this area, and a legume. And if you look at the flowers going by the pollination syndrome concept, it looks like this would be pollinated by birds. But our observation, besides demonstrating that Nectarinia asiatica, or the purple sunbird, can be a suitable pollinator because it has a bill which matches the curvature of which matches with the curvature of the keel of the the flower. But this is the only opening. I have dissected the flower just to show the internal structure, and there is only one opening of the flower, and from where the bird is able to insert the beak and collect nectar from the bottom. And but besides, my observation in the field also showed that sometimes syndrome concept can be misleading, and you may find something which does not belong to that syndrome at all, and your pollinator can be from a different functional group altogether. And then what we demonstrated, my observation also showed that besides the birds, the squirrels were also able to pollinate. If you focus on this insect, you can see the pollen grains on the head region, and this when comes in contact with the flower. So the interesting thing about this plant is that birds are effective in doing cross pollination, whereas the squirrels are more involved in doing selfing. And both these animals are territorial in nature at this time of flowering because this is the breeding season, and that's why squirrel is not moving away from the tree more than 3-4 trees, whereas the birds can move many kilometers and bring pollen from different areas, and that's how they are able to effect outbreeding. So what you are actually observing in the outcome is not only outbred progeny or only the self progeny, but a mixture of both. So that much of variability is there, and now the incidence of mixed mating is increasing with detailed studies on different species in the world. This is another species which we worked on in Rajasthan. This is called Tecomella undulata, or the teak of the desert, and this is a member of Bignoniaceae. Bignoniaceae is actually known for late-acting self-incompatibility. That means it's an obligate system where 100% selfing would be failed. Only outbreeding is possible. So what we started doing when we first observed, what's happening in the population, I came across this same bird, which is a pollinator of Butea. But in this case, this is not the pollinator of this species. Here it is a robber. If you look at the beak, it is pointed very sharp. This is a male juvenile, which is just the breeding season, during when the plant comes to bloom. And it is involved in, taking away the, nectar, and it drops the plant of the nectar, and it is the first one to come. And usually in more than 99% cases, it makes a hole in the tube and takes away the reward. Or maybe it is using the beak like a straw and collecting nectar from the bottle opening. But the interesting thing here is the essential organs are not coming in contact with the animal at all. So they are the robbers. But the pollinators are the bulbul species, and they are the one which have perfect matching of the morphotypes, where the essential organs are then able to come in contact with the nape or the crown of the bird, and they transfer pollen grains. But their visit is after the sunbirds visit the plant and drop the nectar, and the requirement of the bird is not fulfilled. Since the plant is outbreeding type, so it mandatorily requires the pollen grains from different genotypes, and when the plant is robbed of nectar, it forces the pollinator to visit more number of tree species or individuals in the population, and this increases the pollen flow and the fruit set is better. So robbing in this plant does not have a negative effect, it has a positive effect, and that leads to fruit set, increased fruit set because of robbing. So this is the question that we addressed, means how it affects the natural fruit set, and we did several experiments where we made the plant to manually we robbed it, and in one case we prevented it from robbing, and we came with the results. I'm not going into all the details, but the last one is more important, that robbing leads to increased number of visits on the trees within a population, and that increases the gene flow. So it's a very good observation that we came across, and it got published in a good journal. This is another tree from Aravali, and this is called Creteva adansonii, but it is wrongly, sometimes described as Barna. Barna is actually Creteva magna, which grows in riverine areas. And this plant species, if you see isolated trees, you will see the fruit set only in the lower part of the canopy. So apparently one can say that the plant is self-compatible, but the pollinator is missing in this case. The pollinator should be a moth species, and which is not there. Although we observed honeybees, visiting the flowers during the daytime, but they won't visit the stigma at all. They will be only looking at the pollen grains, collecting them and going away. And nectar is also there to some extent. So what happens is, what are these bees doing? That these bees, when they move in the flower, they are actually touching the stamens like strings of a musical instrument, and when this happens, it sprinkles the pollen grains, and with gravity only the lower part of the canopy collects the pollen grains, and the fruit set is there. So that's why the title of the paper was insect facilitated wind pollination. It is not direct pollination, but the true pollinator is missing from the population. This is another endangered species that we worked on, is called Oroxylum indicum, and if you look at, this is another Bignoniaceae member. Late acting self-incompatibility is there, and you can see the inflorescence which is coming out. This is a syndrome typical of bat pollination. Whether you have chandelier-like inflorescence or a popping up one, then only bat pollination is possible. And this requires lock and key mechanism, then only pollination is effected. The major reproductive constraint in this species is the stigma exhibit opening and closing when you touch it. The moment you touch the stigma, it will close and it will never open, even if the pollen grains have not been deposited. So imagine bat coming without pollen grain and there is no fruit set. So this was the major constraint that we identified, and this was reported in a very good journal again. Most of the sapendaceous, taxa have the problem of sex lability. All the tree species, what they do is, some trees to begin with would be male, and the later part of the flowering season they would become female. So that is what is known as sex lability, and this is seen in one of the, this is a very important tree species of India, which is called Schleichera oleosa, and kusumi we call it, which is used for growing lark insects. And the other one is Butea rangani, which is grown on them. So that context, this is a host of a very important insect, which has a product-oriented, perspective, so needs conservation from a different point of view. And the trees are only grown in, I think, Ranchi, where lark production is maximum. Interestingly, some tree species that we found were not pollinated by bird, bees, and anything else, but flies, because the flowers, they emit fragrance which is of fetid type, and they don't smell well. But the constraint there is not that the trees are low in number. The constraint is the density. If the trees are not growing close by, the movement of the fly is not there, and if they are too sparse, the fruit set is poorer. So this has been seen not only in one of the species that we call Serratia numularia. There is another species which is called Serratia, which grows only on Mount Abu and top hill, and that is also pollinated by flies. Now, Professor Yadav was very vocal about not using molecular biology and all that. I am not in favor of that. I think in a multidisciplinary approach, we should also use the molecular tools, because that improves the resolution of your observation, and one should not shy away from doing it. Although I was not trained in molecular biology, but I allow my students to do it. So in doing so, we first used molecular markers to see what is the extent of mixed mating in a particular tree species. This is right here, Tomentosa, and this species was recovered in a sense in the forest through tissue culture. Plants were raised, they were clones, they were distributed and replanted in an area called Kevadegi Nal in Udaipur. And this work has been done by, I think, G.S. Shekhawat from Jodhpur, and is probably one of his friends. Now, trees have now reached the age of reproduction, and we were able to show that. So you look at the flower. Flower has a very interesting, architecture. The stigma and the, if you look at this thing, they are enclosed within the structured dome. And it is not possible to actually collect the pollen grains unless you have a suitable pollinator. But there is an opening between the anthers from the base where the proboscis can be inserted. But when it comes out, it requires kind of an effort. So there are different positions from where the proboscis can come out. So you can take the pollen grain, lodge the pollen grain onto the stigma, while coming out, collect more pollen grains from different angles, and in one go, both pollination and pollen collection can be done. So this is what is happening in this case. So we found that this hawk moth is able to do pollination and brings pollen grain from from a distant source. But you have another set of, moth species which are of settling type, not hovering type, and they have different consequences. They are more of, which brings out geitonogamy, or what we call selfing to some extent, but this one is involved in outcrossing. So they have different consequences, but when we did, our usual experiments of finding the breeding system, the cross-pollination always does better, and there is very high inbreeding depression because when you do selfing, the fruit set comes down. It can happen at the fruit set level, seed set level, or the seed weight level that we observed. So what we did, we used the AFLP marker at the dominant one and used the MLTR analysis. In this analysis, we came across that the outcrossing index is nearly this one, TM, is around 0.68 or 68%, mixed mating is there. So this is the way to demonstrate the relative contribution of selfing and outcrossing when you want to show what is the extent of mixed mating. And mixed mating can be demonstrated not only through hand pollination, but you have to use a molecular marker to demonstrate it. That's why I was not agreeing with Professor Yadav to some extent. Now, this gives a lot of resolution to your observation and acceptability in the world that what you are saying is actually true. Similarly, I'll quickly go through it. Neem is 100% self-incompatible, and then your bale is self-compatible, but large number of fruits are formed, but it's the resource, distribution among the fruits which actually curtails or brings down the extent of fruit set in the tree species, and it is, visited by a large number of insects, but Apis dorsata is the main pollinator. Okay, we did, this is some, now few slides are left. We did some kind of a meta-analysis. This is for the Rajasthan, and we collected, we marked certain species, tree species, and looked at the pollinators. So, and plus we added some of the tree species which have been recently investigated through the All India Coordinated Project. A majority of the plant species, the tree species, are self-incompatible to the greater extent, but many of them are, mixed mating type. And these need to be, validated through molecular markers. And of course, some allow self-compatibility to operate within the system as well. What he showed at the beginning, Professor Daphne, as a plant-pollinator interaction. If this is with respect to the, phenology. We have different months here, and what are the species blooming, and this is the modularity for that. And what it shows that nearly 31 tree species, they bloom during March, April, and May. Large number of them. And then we have other species which, bloom during July, September, and August. So if you go to Rajasthan, that's the best time to visit. But, maximum flowering is happening during February, Jan, July. This is a bigger module. So wherever the node is bigger, you can see the number of individuals or the larger number of trees which are species which are, in bloom. On the left panel is the number of tree species which have been coded, given a different code. Okay, so this is the interaction. This is what we call it bipartite interaction. This is for the plant-pollinator interaction for all these trees. On the top you have the different species of insects, and at the bottom you have different species of the trees that we use for the meta-analysis. And wherever you see that the node size is increasing, they are more like journalists, and wherever you see that the node is 1-to-1, they are more specialist. So you have very few pollinators for one plant, whereas there are a large number of individual trees which have, large number of insects which are doing the pollination. And this is the modularity which shows that for all these species that we put into meta-analysis, Apis is the major genus of insects which brings about pollination in the species. But then you have birds also. Then you have other pollinators also which are important, for which we don't have sufficient amount of information. And if you look at the importance, we see Apis, Floria, Serena indica, Dorsetta, and Mellifera doing pollination for majority of the tree species. But then we also came across, surprisingly, Ceratina is also doing the job. So what I'm trying to emphasize here is not only the professional bees which are known all over the world for pollination work are important, you have the importance of native pollinators also there. So now with increasing climate change, decline in pollinator diversity that you might be reading about in different journals, you will come across a lot of emphasis on now how to save the native pollinators and bring them into exercise. So very beautiful studies, but you may ask me as if in the form of a question. I wish I had more time, but I'd like to stick to 20-25 minutes to keep up and respect. Finish. This is my last slide. So we have polysters also. So this is my second last slide. So what a conservation biologist should do, my observation and conclusion is not different from what Professor Daphne said, that instead of you can keep on playing Darwin by looking for the pollinator of a species, but at the same time, if your objective is to conserve species, then when we say reproductive biology, we are not doing reproductive botany. We are not doing reproductive zoology. It's biology. Then you have to conserve both plants as well as pollinators. So you need a habitat approach, and you need to strengthen the demography of the journalist pollinators, because they are in the network which gives more strength to the pollination network. Then also there has to be an interplay of, if you need to expand the study at the community level, not at the species level. And we are able to publish with one paper and show, look, we have studied reproductive biology. That alone will not conserve the species unless we study all the species occurring in that area and then bring about, look, this insect is able to visit more than 5 plants. All these 5 plants are catered by one insect. So you need to conserve the insect to conserve those 5 plant species. This is the conclusion. And Then you have the recovery program. Then I also have seen that why pollination failure happens, that either it is because of the loss of pollinator or it can be because of the mate. If sufficient number of mates are not there, and if you happen to see that the most of the tree species are outbreeding type, then you need to have different genotypes in the population. There is something called biparental inbreeding. That means two related individuals, if you cross, the fruit set will not be good. So you need to have a distinct individual to have a good fruit set. So when you are recovering species, then you must bring different genetically unrelated individuals in the population. Not like what happened in Tomentosa, where from a tissue culture plant, clones are grown within the same vicinity, and then what future is going to have is that species will not be able to produce much of the seed in time to come. So you need to establish heterogeneity. This is my last important slide, and you can see this is my group which has contributed so far. My funding has come from different sources. These are some of the glamorized things, the journals that I'm showing I published here and also, but yes, they are important to me and to my student. Thank you very much. I can take a couple of questions.
So I have one person, if you can ask question, anybody, one question only. Yes, yes.
Thank you, Professor Rajesh. Excellent talk. I was just wondering, you are talking only about insects or birds pollination. In tree system, you also have other mode of pollination. Are you pitching on that, like air or wind or water?
90% of the species pollination, if you see, is by insects, entomophily, or I would say zoophily. But dioecious taxa, where you have different individuals and sexes, most important ecological correlate there is wind pollination. For example, sea buckthorn, Leh Ladakh, populus, salix, they all grow in a place where they are desert, and there the insect population is not much there. Wind is more prevalent, is an open habitat, and their wind pollination comes into the picture. In Kreteva, we just showed a different story altogether, but it is not exactly wind pollination. All these plants which have wind pollinated traits, they usually grow in such open habitats. But if you go to the denser parts of the tropics throughout the world, it's mostly the zoophily which comes into picture and insects comes into picture. That's all I would like to say. So I've been working on sea buckthorn for the past 10-15 years. That presentation I'm not giving here.
Okay, sir. Thank you very much for nice. So next presenter, Dr. P. Girdhar. Have you given your presentation? So he's from Plant Cell Biotechnology, CSIR, Central Food Technological Research Institute, Mysuru. The topic of his deliberation is conservation of Decalipus hamiltoni, an endangered twiner with modern relevance in health food. There is actually mistyping. So, but anyway, about Dr. Girdhar in 1996. After that, he was appointed lecturer in microbiology in Kakatiya University. After that, in 1999, he joined as scientist in CSIR, CFTRI, and he went for postdoctoral to Germany in 1995-96. He is member of many research societies, scientific bodies, and he got many awards. Srimati Kamala Devi and Sri Ramlal Diman Gold Medal of Academy of Plant Sciences in 1999. Then he was awarded Y.S. Murthy Young Botanist Award of Botanical Society of India 2007. Then he was awarded Professor H.C. Dubey Outstanding Young Scientist Award of Indian Society of Mycology and Plant Pathology in 2008. Elected member of National Academy of Sciences, India in 2008. He was elected member of Plant Tissue Culture Association of India in 2009. He was given Best Research Scientist Award of CSIR CFTRI in 2012. Then so many awards are there, I am not going to read them, but now presently he is Principal Chief Scientist, Plant Cell Biotechnology Department, CSIR Central Food Research Technology Institute, CFTRI Mysuru. Now I will not take much time and request Professor Girdhar to just give his talk.
Good evening to all of you. So it's my pleasure to be here to give a glimpse on my almost 15 years work on this endangered plant species that is called Decalipus hamiltoni, otherwise it's known as a swallowroot. So I will give an abstract form of various aspects that we have covered on this plant during the last 1.5 decade. So you can see this plant is highly an endemic and endangered plant to the southern peninsula of India. So especially it is located in the hilly terrains of Tirumala regions of the Andhra Pradesh and Karnataka, Bihar hills and Wayanad hills of Kerala and surrounding areas, even in Tamil Nadu certain portions also. So the beauty of this plant is it is a medicine, it's like a woody climber and a twiner. It starts its rooting in the rocky crevices, and the twiner it reaches to the canopy of the surrounding plants, and you can see here this plant, this entire one is this Decalipus canopy. So generally it is highly associated with the highly terrain regions. So getting access to this one is one of the toughest thing. So there are other species of Decalipus like Decalipus arealpatra, Decalipus salsiflora, Decalipus nervosa, but the most popular one and common one is the Decalipus hamiltoni plant. So this is the overall picture of different questions of this plant. This is the complete bushy appearance of this one, and this is the floral part, and this is the fruit capsules, and the seeds, each capsule contains 40 to 60 seeds in its locules. So they are very papery and having a feathery structure, so easily they can be disseminated. And this is the complete uprooted plant. It contains long tuberous roots. These are the dry tubers, and this is the one actually you can see how the plant gets started in the rocky crevices of this hilly terrain region. The beauty of this plant is there is one, it is very familiar for its vanillin flavor pleasant metabolites. Its structure is 2-hydroxy-4-methoxy-benzaldehyde with a molecular weight of 152. If it is structurally the same molecular weight compound is vanillin, it is 4-hydroxy-3-methoxy-benzaldehyde. So structurally it is same, only this functional group position is different. But in vanilla, unless you cure the beans for a certain period, you will not get the vanillin flavor. But in this one, naturally, if you prick the tuber as such, it will give the complete vanillin flavor to the entire room also. So that is the beauty of this unique nature of this tuber. So traditionally it is known for its stimulate nature, it relieves flatulence, and also can be used as a general tonic by the tribal communities, Soliga tribal communities and Yanadi tribal communities in the Andhra and Karnataka regions. So it is a folklore medicine. It is used as a good appetizer, blood purifier. So various properties are attributed to this one, but there was no scientific evidence, substantial information available on this plant. That's why in 2000 we got a DST project to explore all these things, especially first to sustainable cultivation development. We have got a project. Subsequently we continued under various mission mode projects of CSIR like Prime Minister's project, Bioprospect project, also we continued on this one. So the root is called, it is used for treating the fever by these tribal communities, and the fresh tubers are sold in the market for 400 rupees, but in off season even 900 rupees it costs in the local markets. Whatever may be the thing, whenever these tribal people wants to bring the product to the market, it has to completely removed from the habitat. That destructive harvestation method is the most common method they use. So as I mentioned, this flavor compound is the major compound among the volatile fractions of this one, that is accumulates to 96%. Subsequently, our institute has studied various aspects of this functional attributes of this plant, like antimicrobial properties, antioxidant properties, anti-atherosclerotic properties, anti-ulcer, anti-diabetic, like that, even how it protects the inflammation related things, and cytoprotective and gastroprotective and hepatoprotective, and most recently neuroprotective aspects also. So this is the beauty of this entire one. It is not alone attributed to the one metabolite, that is 2-hydroxy-4-methoxybenzolate. It is known for its antioxidant nature, but there are other phenolics and flavonoid compounds present that we have identified, that I am not going to present here. So because of this threatened nature due to the destructive harvestation method, this plant became an endangered plant and as per the IUCN database, and also as per NBA, National Biodiversity Authority of reports, it is declared as a species with high concern, this one. So accordingly, we have followed different methods, with both the traditional methods by collecting the seeds from the forest, and also through the alternative biotechnological methods. Way back in 2000 year, to get 1 or 2 capsules of fruits, we used to travel to the BR Hills or Tirumala Hills with a lot of difficulty. From the canopy, we have to collect the fruits and get that one. Like that, we have started. So now, during the end, I will show you the wonder how it has done now, from how the plant has moved from the hilly terrains to the plains with the tissue culture technology and other traditional methods. So this is the tissue culture, various mass multiplication methods actually we have developed, and process technology also has been developed under in vitro conditions, and later we have taken these plants to the BR Hills natural habitat, and how do they perform with the other traditional naturally grown plants. So those studies we have studied, and we have transferred those plants to the Vivekananda Girijal Kalyan Kendra in the BR Hills region. Alternative, since if farmers are using only destructive harvesting methods to get this flavor compound or flavored tubers, is there any possibility of biotechnological methods application to develop the same flavor compound under in vitro conditions? So that's why we have developed the root-based technology. So by you harvesting the roots under in vitro conditions, you can get the metabolite, and also through callus suspension cultures, and also through encapsulation immobilizer studies also, we could able to produce this flavor compound. Suppose in the normal callus suspension, 0.08 milligrams per 100 grams dry weight, you will get it. In vitro routes, 0.24 milligrams you will be there. Upon precursor feeding, that means biotransformation studies, so you can get 0.44, and this elicitor treatments, 10.3 milligrams. That is the beauty of applying abiotic and biotic elicitors to augment the flavor compound in these tubers, like any other biotechnological methods and any other commercially grown plants like soybean, coffee, and other things that I am not going to present here. So this is ultimately we have developed a process technology for large scale multiplication of this plant. Since this is a 2-hydroxy-4-methoxy-vanillin related compound, obviously it is the product of phenylpropanide biosynthetic pathway, the most important secondary metabolite pathway after this shikimic acid pathway. So phenylalanine is the precursor. So as of now, there is no information available about the biosynthetic pathway of this flavor compound. Vanillin, we know it is a phenyl. Similarly, since it is structurally similar, how it will work. So we want to identify the key enzyme involved in this biosynthetic pathway. So by using the certain enzyme inhibitors like MDCA, propanylic acid, we could able to identify the conversion step between the ferulic acid to the vanillin, that is a putative vanillin synthase is one important thing, and there is an unknown step between ferulyl CoA to 2-hydroxy-4-methoxybenzaldehyde. This had to be identified. So we could able to deduce this critical step is the ferulic acid. So we have added ferulic acid to the culture medium, and many folds improvement or conversion of this ferulic acid to the vanillin flavor compounds we could able to demonstrate. So subsequently we have identified putative vanillin synthase enzyme and amplified, and we could able to deduce its partial sequence also, and how the relative expression of the phenylpropanide pathway genes occurs during the developmental stages of this tuber, because you have to wait minimum 1.5 years to get the complete flavor compound and maximum content also. So we could able to track 3 different stages, right from the 6 months, 10 months, like 18 months, and we have demonstrated the active expression of these phenylpropanide pathway genes in the stage 2, that is a crucial step. Later on it gets stabilized. So of late we have developed the neuroprotein toxicity prevention potential of this decalpus Hamiltonian swallow root extract. This we have demonstrated the model organisms like C. elegans. So if you give the hydroxy dopamine treatment, there are it disrupts the neural signals of motor functions in the C. elegans. But if you substitute the decalpus extract, all these gates will be rectified, and again it will functional locomotor acted, physiological behavior, mitochondrial functionality, especially electron transport network, all these things will be rectified. So this is the beauty of this neurotoxin. For the first time we have demonstrated. And another thing is of late we are feeling lot of problems with the prostate cancer. A number of medicines are there. So our recent study has shown the protective nature against this prostate cancer cell lines. So this decalpus Hamiltonian root extract has shown 80% activity against the prostate cancer cell lines inhibition also. Based on this our scientific evidences, hepatotoxic nature and other things, moreover the flavor compound is very important from food technology perspective. Otherwise most of the food products what we use wherein vanillin is absolutely synthetic vanillin, because synthetic vanillin you may get for 5 rupees in a sugar coated powder, but if it is the vanillin bean you have to buy for 300 to 400 rupees for one bean. So a process technology we have developed wherein it is a flavor essence. It is more like a functional food concentrate actually. Few drops you can add to any food products. So for this we have developed in our CFTR itself. These are various steps involved in developing this. Ultimately through pasteurization you can seal, and one bottle cost around 200 rupees. So you can happily keep it in a freeze side door for at least 1 to 2 months time. Whenever you want you can add few drops that one. So because by knowing, then we have developed its application in various food products like beverages, ready to serve food products. This is the spray dried powder of this decalpus Hamiltonian, and in bread and muffins and flavored milk also you can use. So after demonstrating these technologies, the farmers in Karnataka has got interest and they approached as that I can tell you later. So these are the various potential R&D areas associated with this endangered plant species. We have almost 5-6 patents on this one, including one US and EU patent on this one, and more than 25-30 publications on various aspects. So it is biosynthetic pathway elucidation and micropropagation, bioprocessing, metabolic engineering aspect of, and botanicals and cosmetic, especially the insecticidal properties to get rid of the cytophilus and other storage pests associated with the chickpea and other things. Against also it is very effective. That aspect, food technology and bioenergy, especially endophytes also we have applied, and we have shown their efficient role in improving this functional role. So what about its organized cultivation? So as of now I have talked about the collection of the material and demonstrating its functional attributes. But to have a continuous sustained technologies, continuous production of the material is very important. So now the organized cultivation has come to that one. There is one farmer called Mr. Surya Narayan Raju. He is one of the prospective farmer from Karnataka region. He has got many awards from the Karnataka government, and especially he used to cultivate Goa and Mosambik, oranges and pomegranate, and he used to export to the many countries also. But after hearing the technology from the CFTRA, he got interested in this one, and he understood the potential economic benefits associated with this one, because there is one drink called Nannari drink. It is very famous in summer season in Andhra, Karnataka and Tamil Nadu regions. In a crude way they prepare that one by using the hemidesmus and alternate to the decalpus also. So because of that economic potential, he came to us and he bought our technology for 2 lakhs rupees for that flavoring, and he has used his own network of collecting the seeds now through forest and tribal communities. So 30,000 rupees you have to spend to obtain 1 kilogram of seeds from these tribal people. So different networks through forest department. So these are the seeds for initially have to put, and they have raised the bed in the poly house itself, or these are the different seedlings developed in a plastic trays within the poly house, and or otherwise you can directly go to the field and there itself you can make the seed beds like this. Then you can transplant after 6 months to the complete different fields, or you can go for the mulching based of treatment. These are all the photographs from the Ballari region of the Karnataka. So now this farmer is going in 100 acres, this one. He has removed all his horticultural orchids and he is growing these plants. So this is after 2 years, the plant growth will be like that. It spreads. Since it is an accelerator and a twiner, all these twines get spreaded on the floor itself. So this is the 3.5 year, now 3.5 years actually. So you can see the capsules also. So last season he has harvested, after 3 years he has harvested the complete tubers. As here you can see the tuber. So whenever you want, you cannot just pluck the plant. These roots are spread like for all directions. So you have to plow the soil and remove. So each one plant may give 5 to 10 kilograms of tubers. Fresh weight basis. If it is dry weight basis, maybe 2 to 3 kilograms. So he has harvested 10 acres of these plants after 3 years by using this JCB and all the things, and he sold it 1.5 crores he earned. You imagine. So that is the beauty of this one. But this entire market, yes, but the entire market is in an unorganized sector. For the first time, this endangered plant has come to the land. I mean, this one, he, the person, Mr. Surya Narayan Raju, I told you. So these are the tubers in his farmhouse, what he has harvested. And the tubers are kept it in open things, and he can dry it, they will cut it into pieces and dry in open fields. Then these dried ones, they will sell it to the market for this flower production, extraction, or different purposes. Now illegally it is going to abroad also for various pharmaceutical approaches also. So after reaching to that level, the Karnataka Biodiversity Authority and Karnataka Medicinal Board Association, they have noticed this one. They have organized one workshop in the Ballari to give the knowledge dissemination to the farmers. Now the farmers are also approaching this person to now it has spread it to the 200 acres on a contract basis also. So these are all the senior conservator of forests in Karnataka, deputy conservator of forests, all these people, they have personally visited and appreciated that field. So if you look at the economics, the cost of the seeds, as I mentioned, is 30,000 per kilogram, and number of plants per acre is 10,000 to 15,000 you can plant as per the information given by that farmer. And investment cost initially per acre is 2 lakhs. Excluding the land cost, and tubers yield will be 20 to 30 tons fresh weight, or 3 to 5 tons by dry weight, and selling price will be 450 to 550 kilograms. So this is about this one, and this has got wide publicity in local publications, newspapers of Karnataka and Andhra Pradesh. And this is about, in brief, in abstract form, about the biotechnological aspect, that is restoration, multiplication and realization of economic potential of decalipase Hamiltonine. And still a lot of work is happening in our institute on other aspect. So this project has been funded by the CSIR Bioprospects, that is CSIR Mission Mode project, and also of course by CRB and DST initially. So it is not alone my work, a couple of scientists are involved, two technicians are involved in developing the product, and 3 PhD students also involved in this work. And it has brought a lot of fame to CFTRA because now the endangered plant has landed ultimately in the land. Commercial cultivation is also happening. Thank you very much for giving me this opportunity.
Any one question?
Question, what are the other Asclepiads which you think of going for such a cultivation? You know, Leptadenia reticulata.
Yes, Jevanti.
They cost about I don't remember exact figure, but 800 rupees kilo.
Yes, sir. Yes.
So that is another plant which grows in a dry region.
Yeah, today morning only I got a clue on this one. I got interested because one boy is presented in the Children's Science Congress. He has used that Jevanti leaf and he has developed a chocolate, and he is just 5 years, 5th class student, 6th class student. He has demonstrated, he has prepared chocolate and fed to me. He is telling, they claimed that it is improving the eyesight also, sir. Yes.
Yes. Very good.
Yes, yes. It's very good. They are supplied to the Anganwadi children also. It looks nice. Yes, okay. That plant is actually very good. Thank you.
Now I request Professor N.V. Kubalkar sir to felicitate.
Okay. Thank you. Thank you, sir. The next presenter is Professor Partha Pratham Barua from Guwahati University, Guwahati. Dr. Barua did his graduation from Nagaon College and masters from Guwahati University. Under the guidance of Professor C.K. Barua, he completed his doctoral research on vegetation dynamics and animal migration in Kaziranga National Park from Department of Botany, Guwahati University. Actually, he is member of many scientific bodies. He has 27-year-long teaching experience in both undergraduate and postgraduate level. He is member of and fellow of a number of scientific society like International Society for Conservation of Nature and Natural Resources, fellow of International Society for Ecological Communication, fellow of Botanical Society of Assam, life member of Indian Botanical Society, Paleobotanical Society, Association of Plant Taxonomists, Indian Association of Angiosperm Taxonomy, Indian Phycological Society, and is the chief editor of Journal of Advanced Plant Sciences since 2019. He currently holds the post of Chairman, Green Audit Committee of Guwahati University. I request Professor Barua to deliver his talk.
Thank you, Professor Chaturvedi. I am from Assam, as he introduced me. I am on behalf of my team, my colleague, Professor Dandathar Sarva, the Zoology Department, and one of our students, Hunmili Taranpi. With the initiative of the government of Assam, what we are starting in our university to make our Northeast sustainable. My presentation will be a little bit different from the earlier ones. So, what we have seen today, the population has been increasing in world day by day, and from 1990 onwards, if you see the projection from the 2050, there will be a huge number of population will be increased, and to feed all these increased populations, so obviously we need more food, the food of all types. So, with due regards to Professor Chaturvedi sir, we the people of Northeast are strictly non-vegetarian. We are fond of fishes. So our government of Assam took one initiative, just why don't you do something by the initiative to feed the people, because fishes are the lucrative source of the protein for the rural peoples, and in our state, if you go there, their life started with the fish. If you cook dal, you just add in the oil furan, pas furan or furan jeera or like that. But there in our state, we used to give one small fish in the oil, then put the dal, boiled dal. That is furan with a fish. And that is the habit of there. So that's why with the initiative of the government, we are thinking for the aquaculture, and when we search, then we have seen that since 1970, that this aquaculture venture has been increasing, and with the initiative of Food and Agricultural Organization of United States, and record says that from 2018, the requirement was 179 million tons, and it was in 2030, it's supposed to be increased to 204 million tons. So answer is what? That we have to make more food, more fish for nutrition, again I beg pardon from sir, I have to tell the audience what is the nutrition value of the fishes. The fish contains all these things, is protein, amino acid, vitamins, iron, potassium, calcium, selenium, zinc, and specifically the omega-3 fatty acids, which is very essential, particularly for the proper functioning of human health that all of you know, perception development. So that's why what we see, the fish has been used, fish particularly, the fishes available in the oceans are used to prepare the fish meal. This fish meal is not to feed the fishes only, which we used to rear, but that meal is used to feed the fishes in the fisheries, in everywhere, from the coastal region to the freshwater regions, to the piggery and to the poultry, besides the human consumptions. As a result, what happens? As a result, what is happening, the fish stock has been reducing day by day. So if you see these pictures, then you will see then from the 90% stock in 1974, and we are going to lose almost 30%, 70% by the year of 2050 because of this extensive collection of the fishes from different sources. If you see from the demand and cost effect, then you will see the cost and demand of the fish meal is increasing. Since demand of fish meal is increasing, so we are just extracting more fishes from the fisheries. To overcome the situations, obviously, the people have come forward, cities have come forward to take some alternatives. Again, Pandey sir is here. We cannot take food from the rural poor peoples. So there is another logic also there. All these plant products are low digestibility for the fishes, deficiency in essential amino acids, lack of omega-3 fatty acids, then presence of some anti-nutritional factors also. So it needs time. But instead of that, if we think a food algae, which usually take by the fishes, which contains vitamins, minerals, omega-3 fatty acids and proteins, everything, obviously it will be a eco-friendly solution. So that's why with this rationally we started. Then we thought that we have got the informations. We also, all of we know, these algae are not the food. These are 3 algae. There is spirulina, hematococcus, chlorella, and these are the food. Not only these 3, all algae are the food for all these molluscs, rotifer, crustacean, copepos, artemisia, and fishes. It is not there. This aquaculture is a new concept. It was originated in 8,000 years back. It was there in different historical literatures. In China also, we have got literature from 2,000 years old, Chinese literatures. It was reported by Sang in 2004. Today, this microalgal culture has been used as a direct or indirect feed for the cultured aquatic species. There are several studies out there throughout the world. They have used spirulina, chlorella, Sanidesmos, and other things for either to substitute or the partial substitute for the food meal proteins in the diet of various omnivorous and carnivorous species. In China also, they added some low concentration resulting high level PUFA increased weight gains than there. But now question is that what is happening in India, what we are doing in India. If you see the study particularly relating the production of the fish feed is very limited in India. Initiative were taken first by Professor Roma Pal of Calcutta University. She took and she studied the growth, performance and nutritional profiles of some fishes with her students. Shiv Kumar in 2010, 9, 10, 11, he worked on chlorella and formidium and tried to prepare the live feed for the better growth of panaceas. Roshadhuri and Mukherjee, again from the Calcutta University, they used this algae mixed diet, the first larval development in comparison to these conventional feeds. These are the study in the national level. But if I see in the Northeast, only Dr. Papia Ramanujan, Professor Papia Ramanujan, she just retired last year. She had a little work on that. And I was fortunate. Generally, taxonomies are not welcome by any sponsors, DBT, DST, whoever. So, sir knows well. But I was fortunate. So I got the opportunity because of a taxonomist. As you know, it is already recorded that we have 350 fish species are there in Assam, and we have classified it, 3 cultivable fish, sport and game fishes, and ornamental fishes. So, in our biodiversity and aquaculture center, I will show you the next slide there. We have a very good setup that was established by the government of Assam. So that center already successfully developed these protocols of the breeding and larval rearing of almost 16 indigenous species of Brahmaputra basin so far. So that was the booster for us, and that was the confidence for us. And government first tell us, just you proceed, you come work on us, with us, so that we can at least develop protocols for the ornamental fishes. But there was no earlier record for the ornamental fishes. So that's why what we have to think, we have to go for initially, for just I am discussing with you, initially we are selecting one fish which we used to consume in Assam, and as well as it is considered as an ornamental one, this is labio butter. Algae are there. As a taxonomist, I have already reported almost 964 algae from these northeastern regions. That was new to Northeast and Assam. So we selected 2 pristine environments between the Meghalaya and Assam. That is the Karbi Anglang Hill districts, just next to the Shillong Plateau. There is a pristine environment. So there we collected the species. Because all areas are polluted, we don't like to take any risks. So we selected 5 pristine environment, good environment, pure environment, where human interference was very less. And of these 8 locations, we collected algal samples. Maybe some of you may be interested. That's why I am just making this presentation like student friendly. Using morphological identifications for publishing our papers, to establish our things. Yes, we have to go for molecular identification. No alternative for me. So finally, morphologically, we identified 5 chlorophycin algae and 9 cyanobacteria. That is blue-green algae, what we see. Because we are the follower of the Sikachari. So in India, we use the word blue-green algae, not the cyanobacteria. For publication, we have to use the cyanobacteria. Quickly, I'll go ahead. These are the algal photographs because we are maintaining there in our laboratory. So once we identified all the things, because you cannot select each and every species to feed the fishes. So we went for the good parameters for the microalgae. So what is the constraint for the algologists is the production of the biomass. So what we did, first we studied the biomass and biomass productivity and a specific growth rate. And what we see, the specific growth rate is very poor in Carteria in 3.68, and in Westolopsis ramosa, it is very high by begin, but that is the blue-green algae. This is a screening method. Gradually, we proceed for the pigment analysis, because without pigment, you cannot think of algae. So what we see is we have some total chlorophyll A, B, and total what we did. Then we see the chlorococcum contains the highest total chlorophyll, then the calothrix contains the lowest total chlorophyll. So we divided, based on the pigments, we divided these two. Accordingly, we just studied the carotenoid pigments, See the 12, this chlorococum again leading the rule. Since chlorophycean algae do not contain this phycobilly proteins, but still we went for that. We studied phycocyanin, phycoerythrin, phycocyanins. Maybe we'll have some clues for that. So then we studied the proximate analysis for the algae, whether we can use that algae for the feed or not. Then what we see, protein content is high, that is number 7. There is chlorococum. Lipid is high in 3, that is Carteria, both are chlorophycean algae. Gradually we are just tapering our results. Accordingly, same results were happening in the lipid productivity, as and moisture contents also. So then we went for the selection of the microalgae as a feed food. Now the question is which fish we will select. So as government have mandated us, you select one ornamental fishes. But we selected a fish that is called as labiobata, where some past study was here, because without having past records, necessity or requirements, you cannot proceed in a research. So formation of the indoor point of view, biomass containing high protein and lipid is desired, that is okay, already we have studied. And Paul in 2019 and 11, we have got some later research that they already identified what is the requirement for the labiobata, that is the fish. So accordingly, we arrange all nutritional values in the microalgae based on the protein value, and we have sum up all these things, and based on that, we selected 5 green algal species, this Chlamydomonas, Loboclamidis, Chlorococum, Carteria, then Chlamydomonas proteus, to proceed further for preparation of the fish feed. Rest is very simple. This is necessary. Still, I am. Then construction of the phylogenetic trees. Then finally, we identified in both way, morphologically and in a sequencing way, moleculars. Then we went for the compound analysis. With these top 5 species, we went for compound analysis. Through this compound analysis, I will not go in detail. We have got different fatty acids, heterocyclic compounds, alkane, alcohols, all these things we have reported in these compounds. When everything was ready, excluding the essential amino acids, that I will tell you what is the constraint, that is the big constraint with us. Then we collected these labiobata fingerlings from a local source, small fingerlings. We took it to the air containers, transported to our aquaculture and biodiversity centers, where you will see, this is a glimpse of the center, every facility is available. So there we started our experimental diets, preparing, particularly for this labiobata, I will just confine myself to this particular work only. We took 3 diets. One is commercial feed, diet 1, the diet 2, algae-aided feed with composition, algae-aided feed 2 with 2 different composition. These are the compositions with the selected algae. Simply, we just prepared this algal food for it. Here, what we have seen, without having amino acids, fish are reluctant to take the foods. So that's why, particularly in all algae, it is very difficult to finalize which particular amino acid is responsible or maybe will attract the fishes. That's why what we use, what was mentioned in the commercial feed, white amino acids, that we use as a control amino acid, and we did not study it on that. Then we feed these 15 fingerlings with all these 3 diets using multi-tank recirculation systems. Before initiating the feeding experiments, fishes are allowed to acclimatize to the aquarium environments. Then diets were used. Initially, we ate the control feeds. Then we ate, depending on 5% body weight, we ate twice daily at 9 a.m. and 3 p.m. for 90 days. Then diet consumption was monitored and recorded. And all at the termination of the growth trial period, all fishes are weighed after 24 hours of gut evacuation durations. That means for 24 hours you cannot provide them any food. That is a logic and that is the methodology only. So accordingly, we studied the growth performance, the specific growth rate, weight gain fish conversion ratios, then protein efficiency ratio, average daily growth, survivability rate, condition factor. Next, we studied the proximate analysis. And through the proximate and the composition of the diets, what we see? First, initially, we studied the diet. Here we see the protein, AAF2. The protein is high. Similarly, if you see the protein is high in AF1 also, but carbohydrate is high only in CF2, that is a control feed. So with this initiation, we starting this performance of the feed after feeding this algae, because algae is our property. So I allowed them to feed. Just you take. This is my resource. Then if you see the AF2, it is a successfully we have seen, then we have seen the 23 point, from 1.197 boat, 1.1991 to it is increased to 23.17. So with this feed, we can see the body weight how much. I will not go in detail. Similarly, you can see in weight gain also. Yeah. We can go in detail with the weight gains and percentage levels and survival rate, then ADGR, FCPR, these are the specific growth rate, average daily growth, feed conversion ratio, then protein efficiency ratio, everything we see, the AF2 is the best one. So accordingly, we prepared and we think for the proximate composition analysis of the fishes, whether they are improved or not. Already the protein is high in AF2 and AF1, whether after feeding this AF1 or AF2, the fishes are benefited or not. By consuming my algae, whether fishes are benefited, and ultimately we came to the conclusion that our algae provided them high amount of proteins and high amount of lipids every time, in comparison to the control. That is, the control means the commercial feed. So from that point of view, we suggested that we can proceed for production of food by using this 5% and 10% microalgal dietary inclusions with these 5 green algae. Now the constraint is with us. Constraint is with us that first one is, that is a question for you also, that how can we select the amino acids which is compatible for the algae and the fishes? Since no one is there studying in this particular, we have discussed in different institute also. If you have some clues, please provide us. My email ID is there in the abstract. So this is the big questions for us. So that we can use this essential fatty acids is already there. It is improving. But essential amino acid, if we can add that essential amino acids, and if we can identify that essential amino acids which is similar to the fish and the protein, and that will be good for us. Still, we are working on that. Maybe within 6 to 1 year, we'll be able to find it out. But still, we need your help. So this is one piece of work what we are doing in our university. It is a collaborative work of the botany and zoology departments. And now, since at the beginning we collected samples from the pristine environment, but after going through different literatures, then we saw that so many literatures are there where we have seen the spirulina are a good food and feed additive. So that's why we find to search this spirulina, and already out of the 11 spirulina species reported from India, I have already reported 9 from Assam. So it was published in Fedis Repertorium. It was reviewed by Comrade himself. And he suggested some information that this portion you should be incorporated in your paper. That is, I consider it as one of my best paper. So all these spirulina already we have identified, and this arthrospirozygonsia that is reported for the first time from Assam and from the northeast. It was not earlier reported from India also. It was earlier reported as a Arthrospirogyra. This is name sense has been done, Arthrospirogyra, with the advice of this Dr. Comrade. So now what we are doing, we are now targeting all these spirulina to prepare the fish feed for the fishes. Because now the question is mainly what we have seen by working on this, both the part, botany and zoology, fish and algae. Algae are the food for the fishes. I have seen that there is a high demand of the ornamental fishes everywhere. And those who are the lovers of aquarium, they will understand. If you eat some commercial food, the fish, usually fishes lose their color. So to retain the color of the fish, beautiful coloration of the fish, so we need some natural food additive. So that's why I think this such type of interventions is necessary. So where I have got the opportunity to, because of a taxonomist. So thank you very much.
Everybody is having
To the pigmentation improvement in the fish, you can use the
Yeah, already I have done it. Because hematopoiesis contains lots of vitamins.
especially if you use the hematopoiesis formation, the pigmentation, fish also will color and
That will be. But from our government officers, they are reluctant to use to this cultured material from the other sides. So they advise us to depend on this autochthonous word. That's why we use autochthonous means local. They advise us to take the local one. Local means indigenous.
Okay.
Sir, please.
We'll discuss afterward.
Okay, sir. Yes, we need solutions.
Thank you very much.
Now I request Ms. Parma Majumdar ma'am to felicitate Professor P.P. Barua.
You come.
from Plant Molecular Biology Laboratory, Department of Crop Physiology, University of Agricultural Sciences, Bangalore, UASB, GKBK, Bangalore, Karnataka. His topic of deliberation will be endophyte-mediated abiotic stress management in crop plants. Professor Nataraja Karaba, he did MSc Agriculture and PhD. He is professor at the Department of Crop Physiology and head of the Central Instrumentation Facility, University of Agricultural Science, Bangalore. He has worked as the scientist at the Rubber Research Institute of India, Kerala, and the Institute of Wood Science and Technology, Bangalore. He was DBT, this is very important fellowship, DBT, Boy Scouts, postdoctoral fellowship at the Department of Molecular, Cellular and Development Biology, Yale University, USA. He was a visiting scientist at Plant Research International, Wageningen University, Netherlands, University of Groningen, Netherlands, and visiting professor at the University of Saskatchewan, Canada. He is the recipient of the Sir C.V. Raman Young Scientist Award from the Government of Karnataka and the prestigious INSA INSA Teachers Award 2020 from the Indian National Science Academy, New Delhi. He serves as an expert member on the advisory panel of the Department of Science and Technology, Government of India and Government of Karnataka. He served as an Associate Chief Editor of Plant Physiology Report, Springer, and is currently a member of the editorial board of Current Science. Professor Nataraja's major research interest is to understand the molecular basis of plant responses to drought and other abiotic stresses. He has published more than 90 peer-reviewed papers in high-impact factors journal like PNAS USA, Trends in Ecology and Evolution, Phytomedicine, Fungal Diversity, Molecular Breeding, Plant Biotechnology Journal, etc. 10 books, chapter, and edited 2 books he has done. Recently, as a continuation of his major focus on abiotic stress, his team has initiated research to explore endophytes as a potential tool in modulating crop response to abiotic stress. I will not read. He is having very big biodata. He is having many credentials to his credit. But I will request Professor Nataraja to deliver his talk.
Thank you, Professor Chaturvedi ji. I think all the things what you said is not mine, it's ours. Everything is done by my students. I was just serving as an elicitor. I'm continuing to serve as a member in our team. Thank you so much for the kind words, sir, and also for the invitation to deliver a talk on endophyte-mediated abiotic stress management in crops. That's our one of the thematic areas what we have been working. For the benefit of students and beginners, I just want to tell you that I am not a botanist. I'm basically an agriculture scientist from the University of Agriculture Sciences, Bangalore, which is one of the oldest universities in India. We have already, completed 55 years of existence, and I don't want to go into the details, but it has long history, and we have our graduates doing exceptionally well in different parts of the world. And we actually take care of certain activities in the university, mainly teaching, research, as well as agriculture extension, what we call. And our lab is interested mainly on abiotic stress because we have to take care of the farmers' need of dryland crops or the requirement of dryland farmers. So in other words, we pitch mainly on dryland agriculture at the Department of Crop Physiology, that is in the North Block. Last year we hosted, or the last Congress was hosted in our center, and some of you would have visited our campus. It's a beautiful campus. We call it as Gandhi Krishi Vijnan Kendra. So today's story, no lesson. I will try to finish within 15 minutes, but I know that everybody is very tired and you are free to sleep, but I don't allow you to snore. Yeah, so sleeping is permitted, snoring is prohibited. You know why? It might disturb neighbor's sleep. So that's the reason snoring is prohibited. Sleeping is permitted. So what I want to tell you is the storyline. You all know that we have a mandate to have climate resilient crop. We need to produce more food. And in our department, we have interest to understand the mechanism of plants so that we can grow them under this climate distress. And we look at the plant characters. We call them as traits. We have some traits as constitutive. It is already there. Some of them are induced upon induction of a stress, in case drought or salinity or whatever the stress. So most often we feel that certain traits can be induced with the help of elicitors. And our lab has main interest in drought. We have been working since many years on drought research, because we all know that water is there everywhere, but it is not there when it is required. So fresh water required for irrigation and multiple other things. So in other words, under stressful situation, we are asking the question, how can you activate the traits which you have studied, constitute and induce traits? I'm not talking about that part of the work, but we are thinking how to induce these characters which are already there in the plant so that we can activate so-called processes, basic physiological processes, including photosynthesis. Basically, I'm a crop physiologist, and morphogenesis and development events. That's what we are thinking about. So we are looking for option to activate the process, basically using the elicitors. So in today's talk, I'll be talking, microorganisms, or we call them as endophytes, as an elicitors. So we started working since 5 years or so on this particular concept of eliciting the physiological process using endophytes. So we have some good stories, and this storyline in fact started based on the ideas from my collaborators as well as from the animal models. So Professor Yadav and many people are talking since morning that we have to merge with the different disciplines of science to make some story out of it. So this is just to tell you that if you see microbiome research happening globally, people started arguing that if you take microbiome from thin or fat person and make these animal system or model system to eat, the microbiome that is actually given by the lean person will have lean phenotype in the animal model system and fat. This is not my story, it's already published by many people. So we borrow this kind of obesity and depends upon the gut flora. That's what they talk about it. So based on that, we started thinking about the reference is here, that is missing there. I'm sorry, it's not probably projected there. But then there is huge interest now that these microorganisms activate certain characters in animal model and they change the phenotype, what you call outward or appearance or physiological process. So from that context, we started looking at endophytes. Endophytes are the organisms which live within the intercellular space, what you call epiplast. They don't bother the plant the way we think. They are different types of endophytes. I have a lot of collaborators to help me to think because I don't work on microorganisms. I only look at the plant. But we have no idea and data to argue that they actually occupy intercellular space, and different type of microorganisms occupy different zones and they communicate with the plant cell. They talk each other, they knock the plant cell and communicate, activate physiological process. So we have this kind of information now being emerged or generated. And if you see in the literature, this morning I gave this hit endophyte. You see how many papers? 1641, 2022. That means it is highly emerging field. And I actually gave a hit for endophytic fungi, which we are interested right now. See 1165. That means everybody is focusing in this direction to activate the process which are already not activated in plants. So endophytes are essential. That's what we wanted to prove, and we have been proving that along with our colleagues. So to give a small story, so you have systemic fungicide in agriculture universities. We keep using this to kill the pathogen. So when I use the systemic fungicide, or our students, when I use systematic fungicide, you see the compromised growth. This is a Bavestin. And if I now pull out the endophyte and put it into this, actually I make them to grow normal. Not fully normal, but you can regain the growth. What does it mean? Simply endophyte is coming from, or endophyte is erasing, can actually compromise the growth and development or seedling vigor, but I can put it back and I can activate the process. That's all. So I'm not doing anything except putting a consortia of endophytes into it. Since I have a little bit of molecular biology background, we also look at the genes which are getting activated. Some of the housekeeping genes which are there, which are essential for basic physiological process, are suppressed when you treat with Bavestin. That means we are erasing endophytes. Probably that process is also getting disturbed. So that's a message I want to throw you, that endophytes are essential for basic physiological process or cellular process. So seed treatment with systemic fungicide, I think it's a time for review. That's what we have been thinking, and I and my colleague and others, we thought about this. We have Surya Narayanan, who is a well-known mycologist, who is helping us to think in this direction. So we believe that it's time for us to think, if you grow the seeds in a kind of clean environment, there is no need of treating with the systemic fungicide, so that you can actually maintain a seedling vigor. In other words, these endophytes, to some extent, we believe, is transferred vertically from mother plant to next generation. That's how it is, although there could be horizontal transfer. Why this idea we got? Again, from the animal model system. There are nice storyline in this way, that you have microbiome dependent phenotype in child, and they say that microbiome depends upon the type, or your microbiome influenced by your mother. Maternal pool of microbiota plays a very important role in your behavior, including psychological behavior, suicidal tendency, a lot of stories line being done. So mom matters a lot. There is a lot of information being generated in human beings. You have C-section versus normal delivery babies, immune compromised phenotypes, lot of informations are being generated. In the same line, we superimpose this idea into the plant, and we felt, endophytes regulate physiological process, and they are coming from the mother plant. So that is traditional. If you see the local land races or local varieties, you have some kind of mother tissues always there in the seed, and that seed, when it germinates, it picks up the endophyte that is buried inside. But we, crop physiologists, I would say, or breeders, agriculture scientists, we are kind of dirty mind, what we did is we evolved we erased the shattering genes. We have now mechanical harvesting threshing, so we actually collect the naked seed. So the so-called maternal tissues left behind. So you have in domesticated species, shattering gene is not there. So we harvest through mechanical means. That means we actually throw the maternal tissue away, only collect the naked seed. So what does it mean? It means we are actually eliminating the endophyte. That's the idea. And in fact, this storyline is picked up very well. So seed accessory structures serve as a reservoir or maternal pool of microbiota that are required for activating physiological process. So what we propose in one of our recent articles is that threshing yards, what we have, after harvesting we thresh the crop, is basically graveyard for maternally born endophytes. So that's the idea. So I want them to activate physiological process. That's how we started. Then we started thinking aggressively, can we use some of these endophytes to activate physiological process and make sure that they also do better. And one of the ways by which we can do is look for the endophytes which are already there in nature. My collaborator and my colleague Professor Uma Shankar has the habit of traveling all over the world and collecting plant tissue and throwing at us to isolate endophytes. So he has a big library. He developed a big library of endophytic fungi as well as endophytic bacteria. We are now currently pitching on endophytic fungus. So from diverse habitat adapted plants, assumption here is that if it is from salt tolerant plant, if I isolate endophytes which is living in the epiplast or intercellular space, that will have some kind of characters capable of transferring to the host with respect to salt tolerance. Here it could be temperature, here it could be cold tolerance. So that's the idea, and he is actually using this endophyte for multiple other purposes, but I am looking from the angle of manipulating plant physiological process. So I want to use them as an elicitor to activate the physiological process. That's how I look at it. In fact, initially we have series of experiments in laboratory condition to pick up the right endophyte. We have so many different characters and make sure that we isolate the one which having specific trait characters. Say for example, this endophyte is capable of going at 2.5 molar salt concentration in NaCl. That's too much. So like that, So we screen the endophytes. Since the time is very limited, we started asking those endophytes to enter into the crop of our choice. We actually played some tricks to make sure that they enter in this case, example 1, rice. You can see that stress versus control. Wherever there is untreated versus different endophytes, you can see the significant improvement in the growth, seedling growth. Not only one, I will not go into the details. We have several examples now to argue in different crop, both in dicot as well as monocot system. Some of our PhD scholars as well as master students who do their thesis work demonstrated the relevance of these endophytes in modulating plant growth and development. So we actually evolved an approach. We can now isolate endophytes and ask them to colonize in the crop of our choice, and we re-isolate and make sure that they are there inside, and started studying them under laboratory condition as well as field conditions. Say for example, my interest is drought. Now these are all the endophyte colonized, this is non-colonized, and we impose drought at vegetative phase, we impose drought at flowering stage. There is a different characters, regulate the drought tolerance here. Fruit development stage, source sink activity regulates the fruit growth and development. All these things we wanted to ask and question whether these endophytes can do something. So from that angle, we did both laboratory as well as controlled, containment facility experiments and demonstrated that some of the endophytes, I'm not saying all of them doing, some of the endophytes are capable of triggering, activating physiological process, including photosynthesis and contributing for better biomass and yield. What happens in the field, we do not know, but in the controlled environmental condition, we are getting very good, interesting leads. Not only in those horticulture crops, I call it as e-enrichment technology, what we are doing, and we have Indian Council of Agriculture's funded programs going on to test whether these endophytes can also do under field situation. See, in case of rice, this is for salinity stress tolerance. We impose different levels of salt stress. Generally, 4 and above decisiemens per meter is a salt stress. Plants cannot do well. In fact, rice system. So you see a maintenance of photosynthesis, maintenance of overall growth as well as grain yield. I will not go into the details. There are now good examples. I have multiple other crops as well, including pigeon pea and other crops. We have now handled to manipulate the physiological process. So we don't know the mechanism. We have a small activity now, one paper is coming up in this direction, that there is an improvement in photosynthesis. What would be the possible mechanism by which photosynthesis is getting enhanced? We have certain models to test. We have CO2 response curves and, for core models, these kind of data sets, we play with them and try to understand. We realize that endophytes contribute for overall photosynthesis or physiological process by acting as additional sink and also contributing for enhancing intercellular CO2 concentration, suppressing photorespiration, overall suppressing the inhibition of end product. Triose phosphate utilization is enhanced. We will not go into the details. Those who are interested, I am just flashing keywords here. We can talk more on that. So they have a lot of role to play to manipulate plant physiological process. And in fact, some of these crops, you see that without endophyte, the plant is unable to grow under salt stress. In the presence of salt stress, you see a happy, grown plant there. So Pokali rice, which is a salt-tolerant variety, we isolated endophyte from them and insert into IR64, which is a very sensitive variety. So we have not done anything except inserting only one endophyte. So this Pokali rice IR64 started behaving like Pokali. What is the mechanism? We don't know at this point of time, although we have some leads to argue that there are certain, through transcriptome analysis, we have some idea. There is calcium signaling mediated responses. We will not go into the details today, but then ultimately it activated the specific physiological process leading to better performance, not even tip burning. That's a typical symptom in rice when there is excess salinity. And this is one way we are looking at it. And we also have global metabolome analysis where we monitor the hormone levels. We have some kind of hormones, phytohormones. We will not again discuss that part. I do have hormone as well as differential accumulated metabolites identified, which are to be characterized further. So the storyline is very interesting. One of the fungus what we have identified is also sequenced now. We have entire genome of this fungus. We are trying to understand what is the specialty it is doing. But then some of these fungus, what we have, at least 3 now, capable of activating physiological process. That's the bottom line. I want to tell you that. So, based on our leads, we started using Arabidopsis model system to ask the question, what is it is doing? Is there any hormone-mediated response or any electric signal or unknown peptide signal? We do not know. So we started playing with one of the fungus which is capable of colonizing in Arabidopsis model system. If you see the phenotype, this is one of my PhD students right now working. You see the phenotype between these two. This is without fungus, with fungus. We have several fungus not doing anything, but they are colonizing. But this fungus is unique. So we have now handled, we have an options to manipulate the process and understand the mechanism as well. Whatever may be the case, with the help of World Bank funded project, we evolved a concept and also strategy to harvest the endophyte from the habitat adopted plants and characterize them and take them all the way to our crop to activate physiological process. And we also have now option to take them to next generation, that is vertical transmission, what I said, so that if I treat and give to the farmers, probably it can stay with the farmer for a longer period. So this model, sustainable stress management option is available with us now. We are very happy with this kind of progress. So what is the message for the students here? So I see when I was a student, plant phenotype is equal to genotype into environmental interaction. Now we started adding more and more. The behavior of or the phenotype of an individual, or in this case plants or whatever it is, depends upon the associated organisms, microorganisms, as well as environmental organisms which are outside the boundaries of the plant. So this we call it as hologenome. I am not calling, others people call now as hologenome. It's not only plant genome, the microbe associated endophytes or the microbes which are living in the apoplast matters. So they govern the behavior of the, be it be animal or a plant. We are only bothered with plant. So environment and endophyte plays a very important role in throwing the phenotype. We also now know that endophytes occupy different types of occupy different tissues, and they manipulate the process in a different way, because it's like a society. This root cell should communicate to shoot, and here again there is a lot of communication happening. Every cell is talking individually, independently, like a cooperative society. For that you need additional endophytes cooperating. So we have drought tolerant traits, say for example water conservation, we have some identified endophytes, cellular level tolerance to maintain the membrane stability, for example, or maybe extracting more water, we call water mining characters. We have some kind of leads to argue that a specific endophyte can do a specific process, or it can actually add one or more events. So this is what endophyte mediated crop manipulation is the way now we are thinking. So smart crops possible with the help of endophytes. So trait activation, therefore, there is a way for us now to harvest endophytes from stress adapted plants. They are rich source of endophytes. They could be successfully transferred to the crop of our choice. There is a possibility of trait activation, and endophyte mediated crop manipulation is possible. So I don't want to talk more about it. Sustained activity of primary processes can be induced by using endophyte as an elicitors. We have to now we have data to argue that there are stress responsive genes getting activated. There are housekeeping genes which are very important for normal metabolic process under stress getting activated. So endophytes, elicitors, are really good to manipulate the process. We can also give a product which our university and ICR, Indian Council of Agriculture Research, keep asking us. We have an option to do that. So my entire project is supported by, most of the project is supported by ICR as well as Department of Biotechnology. And we have a program under National Higher Education Project supported by World Bank. Some of the work what I presented is from those. I would like to recall the contributions of my collaborator as well as mentor like Professor Umashankar, Ralph from Germany, Surinaran from Winstrom, Chennai, Theo from Groningen University, Netherlands, Ragottama from US, Purdue University, Karen Tenino from Saskatchewan University, and Paranjit Khurana, Delhi University, UDSC. So all these fellows are, in one form or the other associated with our program. I'm very thankful to RTM Nagpur University for giving me an option to share my views. And these are my students. Pallavi, Rachana, Abhishek, Chetana, Kiran, Padmaja, Danyalakshmi, Mansa, Hemapriya, Aisha, and Akshata. A few of them are master students, most of them are PhD scholars. They generate data, all the things whatever chairman read out is their contribution. I am only acting like an elicitor. I would like to thank the president, sectional president Chaturvedi Saab, for giving me an option. Thank you very much.
Thank you, sir. Work very nicely within time, and I request you, sir, take seat. Okay, yes.
Now I request Professor Alka Chaturvedi ma'am to felicitate Professor Nataraja sir.
Nagpur University, Nagpur. Deliberation will be on sustainable development, challenges and biotic intervention. Dr. Chaturvedi did her PhD in taxonomy from Indore University, department in the reproductive biology from the Nagpur University. Then his specialization is angiosperm taxonomy. He was head of the department of Nagpur University. She was head department of molecular biology and genetics. Major activities, teaching and research. She received Best Teacher Research Award, Women Scientist Award of RTM Nagpur University, Professor Sivarajan Award, Savitri Birbal Sahni Award. She published nearly 170 papers in refereed journals. She had 12 funded projects from UGC, MOEF, and Forest Department, University of Bloomingburg, USA. She had the project on park management at Bloomingburg, USA. She guided 23 students for PhD and 20 students for MPhil. Now I request Madam to deliver the talk.
Very good afternoon to all. And before starting, I must thank all the persons who are still here, even at this time. I'm thankful to the chair, to the president of this botany section for giving me this opportunity. It is really very brave act on part of you to remain here after having a visual treat from the earlier speakers. Now I'm going to back to the theme of the Indian Science Congress, Nagpur. Here, sustainable development challenges and biotic intervention. Before starting my topic, I would like to say that what is sustainability? Is it a concept or hypothesis? Legacy or dream? If it is concept, then we have to develop it. If it is a hypothesis, to prove it on the basis of our expertise. If it is a legacy, then we have to protect it. And if it is a dream, then we, with our wide open eyes, we have to materialize it. So basically, I am going to talk only overview of this concept of the sustainable development and how far we botanists can help in the development of this sustainable development. Now regarding this picture, here it is very clear that sustainability is not a unit, it is not unitary in nature, but it is composite and it is having three components, that is society, economics and environment and their complex interactions. There are a number of challenges which are well known to all of us. They may be of climate nature, climate change, food security, preservation of ecosystem services, conservation of threatened species, and control of invasive species. It means from all round there is bombardment for the sustainable development. Climate change, as we all know, we all are suffering from this, and it is affecting all sphere of our life, whether it is health, agriculture, forest, water resources, then bank of the oceans or forest, and even it is forcing existence of the species. Today in the newspapers you might have read that rhododendron is flowering in the northeast region, then Butea monosperma and mango, Mangifera indica is flowering in this season around the central India. Quite early from the usual time, and this shows that it is only because of the climate change that we are seeing this type of the phenomena. But the another picture, just now we have seen so many pictures of the role of pollinator. Flowers may have come to the flowering, but what about the pollinators, their availability and process of the fertilization and seed setting. So in this way, this climate change is having so many effects on general feature of the environment. Then question comes, now we are here as a botanist. What is our expertise? What is our botanical learning by which we can change the picture for the benefit or welfare of the mankind and even environment? As a botanical expertise, we have ability to identify resources, where they are present, what is the identity, what is the stretch, and how can we use this botanical material. We have the expertise of restoration of the habitat, protection of the biodiversity, Number of the services which are performed by the plants, we know about that. So we can take care of the health of human being and environment, and at the same time, we can do the mitigating effect for the challenge, climatic change effect. But question comes, what is our strength? Now, if we see network of the plant scientists, in India, more than 50,000 plants are available, including lower and higher plants. And this network of plant scientists, it includes number of research institutes, number of universities, various type of the universities, colleges, schools, and even NGOs. So entire length and width of the country is having large number of the persons which are trained in the botany, and they know which plant is growing there and what is the significance of that plant. So for botanists, there are certain targets for the sustainable development to promote the circular economy. Earlier, this term circular economy was not in use, but now we say whatever you take from the nature, you have to put it back. So for the sustainability, there is 3R, that is reduce, recycle, and reuse the term. Then second is the integrated and sustainable processing of the biomass. Third is valorization of the bio-waste, and fourth is environmental rehabilitation of those areas which are not in good health. So these are the targets by which we can go to the sustainable development. Now, as far as our nature, our land is concerned, we know that out of the 329 million hectare of the land which is occupied by the India, the country, around 90 million hectare is the wasteland. And surprisingly, 40% of the population, it is depending on this, around 33% of the wasteland. Another feature where we consume that land is the basic natural resource. If natural resource is not in the good shape, how can we use it? Another point is the deterioration or desertation of the land. In this case, erosion of the topsoil, change in the land use pattern, destruction of the habitat, and then invasion by number of the invaders or exotic plants. They have destroyed the land fertility, and there is the botanical expertise by which we can change the nature of the land. We can bring the deserted or wasteland into the potential progressive land. There are a number of the features. For example, cultivation of the plant which can increase the fertility of the land, use of the manure, and then bioreclamation of the degraded land. It is said that if you select plants which are their own plant or native plants which are hardy plants, and if they are having high productivity, so these indigenous plants, they can maintain, they can come over the drought period. Similarly, reforestation of the land, local plant, and likewise creation of infrastructure that is needed for the agro-processing, by which we can give value to the waste material from the agriculture field, and in this way, by this bioremediation method, we can remove the degraded land to the fertile land. In case of the environmental pollution, we say that pollution may be of the air pollution, water pollution, plastic pollution, or soil pollution. When we talk about the soil pollution, we say that it is the degraded land is the polluted land. In case of the air pollution, just few months back or even today, we are seeing that burning of the parali in the Punjab and Haryana is creating problem for the Delhi or North Indian people, and this is basically because of the air pollution. It is reported that from 17 to 25% of the solar yield is stopped, is reduced because of this air pollution. And It is the main cause for number of the diseases which are related to the lung, pulmonary obstruction, or sometimes strokes. They are also counted on because of the air pollution. So what is the best solution to check this air pollution? It is said that plants are only cost-effective alternative. There are a number of sensors, number of the mechanical mechanisms are now developing, but plant is the cheapest resource which can be used to reduce the air pollution. Particularly in case of the industrial area, if we make a broad belt of the green belt around the area, it can reduce the pollution level of that area. Similarly, in the urban areas, having rooftop mechanism or vertical forest, avenues, trees at the crossroad, these are the good example which increase the greenery of the area, and at the same time, not only they are reducing the air pollution, but sound pollution, and they are also giving pleasant to the eyes. Number of the material which are produced by the green technology, like protein fibers, they are used for filtration of the water, purification of water. In addition to number of the plants which are basically aquatic plants, but they have the capacity to reduce the pollutant from the water. There is one interesting example of Ecornia crassipes. It is supposed to be invader plant which is occupying, very fast growing plant occupying entire surface of the water. But when it is limited to a certain area, the same plant is considered as water purifier plants. So in this way we have to select what are the plants which we can use for the purification of the water. Likewise, very recently one plant is identified that belongs to family or group of fungus, mushroom. It has the capacity to break down the plastic material into small molecular material or convert into non-biodegradable material to the organic material. Thus, this can be used for maintaining the landfills where we are just dumping lot of the waste material, including glass, plastic, and large number of the waste material. So by using this mucor, this mushroom, and number of other bacteria, we will be able to decompose all this non-decomposable material and utilize them for benefit of the soil. So we have seen pollution, we have seen land, how to convert non-culturable land into the culturable land. Then question comes, what about the agriculture? India is a very vast country with number of bioclimatic zones, and every climatic zone has its own peculiarities. So question comes, what should be there for the sustainable agriculture? Basically, sustainable agriculture is the organic agriculture where we try to maintain soil, plant and animal as a system, and this system as a holistic approach which affect not only present but future also. Then there is organic fertilization in form of manure, compost, herbaceous plant, leguminous plants, and organic pest management, where number of the plants are used for killing the pests which are affecting agricultural fields. There is fertilizer tree system. Now, system is very old, only nomenclature is new, that is fertilizer tree system, where leguminous trees are used for improving the fertility of the soil. Then rotational woodland. If we alternately cultivate crop with the woodland, there will be more growth of the woody plants for the other purposes, and at the same time, different type of the plants on the same site, it will attract number of the pollinators, and thus they will increase the productivity of the plant. And similarly, fodder bank. Always we think about ourselves, not about our animals, particularly the pet animals, which are improving, which used to improve our economy. So, in case of leguminous plant, if we harvest them, dry them during the rainy season, we can make them useful for these animals during the summer season. There are a number of the plants which are highly rich in protein content, and even they are now available in the markets. Our knowledge in the physiology, in the agriculture field, in the behavior of the plant, in the ecology and physiological aspect of the plant, now we are able to see that these plants are also stressed by number of abiotic factors. How to remove? Just previous to me, there was one lecture on endophytic material as material for check the abiotic stress. So we can manage the soil amendment, inorganic amendment, organic amendment, use of the anti-transparent, use of the mycorrhiza. There are a number of the techniques now available by which we can remove the stress factor of the plant and allow them to flourish. At the same time, one advice is that always choose hardy one, local one, so that they can compete or they can overcome the stress condition. Then comes the food sustainability. It is very sad that recently in the global hunger index, our India's number was 107. Even the smaller countries like Bangladesh, Pakistan or Sri Lanka, they are above in position than us. But where is the problem? If we see the productivity of the land, productivity or food generation is increasing, there is more production, but still food availability is stagnant. The basic thing behind this is loss of the material. We are not able to manage them. We are not able to store them, and there is problem of the marketing. So whatever efforts we are doing, starting from the Green Revolution till today, number of the information technologies and criteria are developed by which we are inducing more food in the market, but in spite of that, there are certain limitations. They may be the cause behind 107 position in the hunger index. Now question comes with the limited space and with the increasing population, and when large portion of the India, large portion of the country is land which is not having that productivity, what we can do? So in that case, zero acreage farming or rooftop or vertical farming, soilless farming, hydroponics, number of the techniques now we are able to see that they may increase the productivity or food outcome. Likewise, use of the wild edible plants, although horticulture or plant breeding, number of the strains are coming, but still wild plants are the best plants because they have grown in that area, they have tolerated all type of the condition, so they are best material for increasing the productivity. Similarly, by biotechnological, nanotechnological aspect, we have seen how to induce, how to augment good character in the material and by fortifying food, and in this way we can remove the hunger, we can remove the weakness of the human being, particularly in India. It is said that India is biotechnologically, not only biotechnologically, but biodiversity rich country, which we have more than 50,000 general plant and around 17 to 18,000 angiosperm plants, and most of these plants we are using. So, if we see what is the accession, these 17,000 plants, they are only identified plants. But what about the accessions? What about the races, different races, eco-races, cyto-races, different type of the races? It is very amazing to see that more than 1 lakh accessions is present for the rice. More than 7,000 accessions are for the wheat. This year is declared as millet year. Why? Because all these small grains, which are available in country, but still we are not using them. We are running for the wheat and rice, which is not our own food. But if we utilize our own food like millet or other small grains, there will be no shortage of the food material, and at the same time, we will be nourished by our local material. These wild accession, they are also useful for improving the crop material. It is said that be vocal, get vocal about local. So, if we see that, if we bring the material from the outside, for example, apple or ananas or any other thing from the other side, it will cost. There will be transportation, there will be use of the fuel, there will be air pollution, and at what stage the material will reach up to the user. So if we use local plant, it will increase the livelihood potential of the local, and at the same time, no air pollution, no shortage of the fuel, and we will get fresh material. So it is always advisable to know about your local biodiversity. What type of the food is used by our tribal people, our local people, or even earlier generation, so that we can popularize them, we can use them. When we talk about the food agriculture, then pollinators cannot remain silent. Pollinators, as we know that 87 major food crops, they are pollinated by the bees. There are variety of the pollinators. And these pollinators, with the climate change, with the change in the land use pattern or habitat destruction, they are also disturbed. They are losing their life. Number of the pollinators, they need protection. They need conservation if you want to improve quality of the food and quantity of the food. All the uses of the plants, whether as mitigator of the pollution, whether as provider of the resources, unless there is conservation, we cannot use. Although plants are said to be inexhaustible source of the material, but if there is no regeneration, if there is no protection, conservation, we cannot use them. And at the same time, when we say that 2 of the 5 plants, they are going to die or they are at the stage of extinction, then question comes how to conserve them for our future utilization. In situ conservation, we know in case of India, 4.7% land is under various type of the protected areas, bioresources, biosphere, wildlife sanctuaries. Then there is new term which is conservation through use. At one hand, we say that overuse is responsible for loss of or extinction of the plant. But if we use them, we will care them for longer use. So this is the new technology or a new term which is coming for the conservation. Then community-based conservation, where we see that this is a recommendation by the international agency that all these national parks, they're supposed to guide, they're supposed to teach and give information to the local community about the plants, about the conservation methods, and about the various uses which comes from the plant. Even a layman, even a man who is not from scientific field now know that whatever oxygen we are taking as through the respiration, it is coming from the plant. Whatever carbon dioxide which we are emitting, it is used by the plant. So this is simple method now. It is only because of the publicity or advice or advocacy by the scientists like plant scientists or members who have placement in this type of the park. Now local people, general people, laymen, all they recognize plant as a great source for our own existence. Then there is joint forest management. Although this is government instrument for involvement of the people for making conservation effort, In addition to this, we know that there are various types of the ex-situ conservation also. Now, ex-situ conservation, they are more needing manpower, more money. But in case of endangered plant, in case of rare plant, where we are not having type of the climate or material where we can use them in vivo condition, then in vitro condition, protection them, conservation them, multiplication them, and then rehabilitation is the good idea for making them future use. Then, after conservation, it is not only oxygen, carbon dioxide, it is not only food item, it is not only mitigator of the pollutant, but number of the products which are coming. The term we use bioprospecting, but bioprospecting is for fuels, although petrol, diesel, which we are using, which is exhaustible, but that is also product of the plant. But from the current plant, from the live plant, how can we use for biofuels, for pharmaceuticals, for cosmetics, personal uses, as biofertilizer, biopesticide, and for veterinary antibiotics. Through the green energy, clean energy, now we are able to develop industrial biotechnology, and through the industrial biotechnology we are able to produce number of the components which are used in the industries like bioethanol, bioenzymes, xylol, bituminous, biochar. So number of the things in general which are used in the industry, in the manufacturing now, which are the outcome of 2G and 3G generation. Likewise, cellulose, which is supposed to be plant product and supposed to be inexhaustible source of raw material. Variously, this material is used in the medical science, in the development of biodigestible, bioplastic, biogas. Number of the items earlier which used to be plastic, now they are through the cellulose or our natural byproducts. Even for the protection of the produces, plant produces, we are using again plant product for saving the life or increasing the shelf life of the plant. Likewise, in the transportation, when one plant material move from one place to the another place, then question comes, how long it will remain in the good condition? So now technology has developed which will give identification that this plant is now in the ripe stage, and if it kept for the long time, then it may go for the spoilage. In case of, just 2 minutes. In case of the healthcare, we know that healthcare, livelihood, and economic sustainability, it depends upon number of the plants. 80% of the population depends upon the plants for their healthcare, and if we see the economy, it is gradually increasing. So this is not only having healthcare, but also economics of the country is increasing by conservation of these plants. And now even the mushroom has been used, or it is supposed to be used for making the computer chip. So sustainability is not only related to the environmental issue, but it is related to the social equity, economic development. It protects our natural environment, our human and ecological health, while driving innovation and not compromising our health or our way of life. So in totality, we can say that sustainable development is necessary for the environment, but for our wealth, our health, and our economic development also. But this is possible only through the innovations in the plant science. Thank you.
I will allow only one question. If no, then thank you very much, ma'am.
Request Professor Ashwajit Fulzule sir to felicitate Professor Alka Chaturvedi ma'am.
Aye, sir. Okay. For nice deliberation. The next speaker of our program is Professor B.P. Mishra, who is professor in the Department of Environment and Environmental Sciences, Mizoram University, acted as head of the department for 3 tenure. Presently acting as Dean, School of Earth Sciences and Natural Resource Management, Mizoram University. Supervised 18 PhD scholars. He had many research projects funded by DST, MOEF, then NRSC, Hyderabad, NIPCO, IJOL, IIRS, Dehradun. Then GB Pant, Himalayan Education, Dehradun, Almora. No, this is, what is it?
Himalayan Institute of Environment.
Okay, this is from Almora. He has published more than 100 research papers in the international and national journals. He is member of several academic societies, subject expert in several organizations and institutions. I request Professor Mishra to deliver his talk.
Thank you, sir. First of all, very good evening to everyone present here, the dignitaries on the dais, esteemed faculty colleagues, research scholars, media persons and others. The earlier speakers, they spoke and covered holistic aspects of the theme. About nothing, a little bit left for me. Something was left. But Professor Chaturvedi just now, she has also covered sustainable development. And I assure you that I will complete my presentation within stipulated time. Please be happy, because it seems that all are tired, including me also. And I am last but one, but my name was last there. So waiting. Now the time has come. One song I remember कि बहुत देर से दर पे आंखें लगी थी हुजूर आते आते बहुत देर कर दी. This is Hindi. So I am not singer, but that line I remember. But altogether, I will complete within stipulated time, and my speech is just interactive to the faculty members and informative to the scholars. Thank you very much. Now I will. How biodiversity is very important. Long back it is written in the Matsya Puran, Das Koop Samavapi, Daswap, entire northeastern India except Assam and Andaman groups of Iceland, then Sundaland includes Nicobar groups of Iceland, and Western Ghat includes entire Western Ghats. Now, causes and consequences of the biodiversity loss. Here I have mentioned natural and anthropogenic. So natural we can't stop. We are responsible for loss of species or destruction of the biodiversity. For all these things, actually we have to think as being the human beings. Anthropogenic overexploitation of genetic resources, genetic processes, many things I have given here. Shifting cultivation, that is very prevalent in the northeast India, especially in the tribal states. Normally there is forest fire burning in the month of February and March for the shifting shifting cultivation slash and burn process, and that leads to the environmental degradation also. Earlier, one line I would like to mention that during Professor P.S. Ramakrishnan period, there was philo period of 20 to 25 years, but now it has been reduced to 3 to 5 years. The people are coming to the same site within 5 years. Because of that very reason, now the shifting cultivation practice is very dangerous from environmental point of view. Now I am coming under the IUCN categories status on the actually decreasing degree of the disturbance. These are the categories. On out of that critically endangered species, we have to save such species, because if in immediate future we are not going for the conservation measure, then that species will be eliminated from the system. I am not going to read that, please. Now, effects of disturbance. What are the effects? I have listed here: loss of biodiversity, forest depletion, land degradation, environmental deterioration, ecological nuisance, and climate change. Now, biodiversity conservation strategies. Madam has already mentioned it, and a little bit I will touch it. In situ conservation within the habitat for the students, and ex situ means outside the habitats. And here we are having biosphere reserves. So till now, 18 biosphere reserves have been notified so far in the India. National park just more than 100, 105. Wildlife sanctuaries 534 like that. Mangroves 34 we are having in the eastern and western coast. Wetlands we are having. Tiger reserves we are having 53 in India. World heritage sites, coral reefs, preservation plots, protected plants and animals, birds protected area. Because I belong to the northeast, basically I am a native of the Varanasi, but I am serving in the Mizoram. So my main focus on the northeast, that their sacred grove. Professor Daphne mentioned about the sacred grove. The sacred grove, actually I would like to mention here, I have extensively studied the sacred groves of Meghalaya, and Meghalaya is very well known for maintaining the sacred grove. And out of the complete forest, 90% community forests are the community forest, and sacred groves ranging from 0.1 hectare to 900 hectare. I have visited more than 100 sacred groves. So what are the sacred groves? The local community, they are maintaining the sacred forest on the religious basis and cultural basis, and conserving such forests. And they believe that Sylvan deity is residing inside those forests, and if anyone is cutting the trees or plugging the flowers or twigs, his or her head will turn down and he or she will die. But due course of time, because of the urbanization, there is decline in such type of religious belief, and most of the sacred groves are facing different degrees of disturbance. Yes, sir? Yes. Most of the sacred groves are facing different degree of disturbance, and during my study, I found that mild disturbance is supporting the high species richness, and when the degree of disturbance is increasing, then if we are talking about the havoc, the trees are replaced by the shrubs, and later on shrubs by the herbs. So now I am going to the next one. Ex situ conservation outside the habitat. We are having botanical gardens, zoos, institutional garden, home gardens. Home garden concept is very popular in the state of Assam. Professor P.P. Barua might be knowing. And arboretum, orchidarium, institutions for germplasm conservation. Now, in situ conservation site, here I have given the objective features and zones of the Biosphere Reserve, National Park and Wildlife Sanctuary. So here I am not reading zones, all the zones, core zone, buffer zone, restoration zone and cultural zone represented by the Biosphere Reserve. In the National Park, we are having only core zone, and Wildlife Sanctuary, core zone, buffer zone and restoration zone. Now, biosphere reserves, no need to read that. Functions of the biosphere reserves. Here developmental functions, we are having conservation functions, we are having the logistic function, so conservation to conserve, and development function, this is related to the socio-economy, and logistic, this is based on the researches, and biosphere reserve. Northeast India, now we are having 8 states. If I am not mistaken, 65% of land covered by the forest. If we are looking at the Indian context, 21 point something. Because of that very region, out of 18, 5 biosphere reserves are lying in the Northeast India. Nokrek biosphere reserve in the Meghalaya, Dibru Sekhoa and Manas in the Assam, Dehang Devang in the Arunachal Pradesh, Kanchenjunga in the Sikkim. And the sacred groves of Meghalaya, they are contributing a lot towards the recognition of the Northeast India under the hot spot of the biodiversity. No need. Now, map biosphere reserve in India. These are the biosphere reserve under the UNESCO program of the map. Now, potential sites for the biosphere reserve, these I have listed here, and presentation will be here for the students. And national parks, no need to read that. Wildlife sanctuaries, number I have already told. Here in the national park, India first national park, this is IUCN category 2, this is Haley National Park, now known as Jim Carvey National Park. Now this is the summary of the currently available protected conservation area in India under different categories I have given here. Okay. Tiger reserves, some details I have given here. Now, coral reefs are nothing but found in the sea due to deposit of the corals and very rich in the biodiversity. Now, wetlands, details given, mangrove, salt-tolerant forest ecosystem situated in the intertidal zone. If we are talking about west coast and east coast, the west coast we are having 16 and west coast we are having 18. List I have not given here. And ex situ conservation site, botanical gardens, definition no need to read, and some important botanical gardens I have listed here. Now, zoos and institutions where several living animals are conserved and exhibited in the captivity. Important zoos in India are these. So why I am moving faster? Because we have already crossed the timing, and this is actually academic gathering to meet with the faculty from other institutions. Yes. This is the main, and Indian institutions involved in the biodiversity conservation, many are there, but these are solely for the biodiversity conservation. National Bureau of Plant Genetic Resource in the Delhi headquarters, National Bureau of Animal Genetic Resource, this is in Solan, Himachal Pradesh, and National Bureau of Fish Genetic Resource in Lucknow, Uttar Pradesh. Establishment here is given here. Now, Biodiversity Act, laws and policies, this is very important. Now the greed is over the need. Because of that very reason, there is over-exploitation of the genetic resources. My need is 1 kg, but if I am going to the garden, I will plug the 5 kg. So why over? So, then actually some legal boundaries should be there. So for that, Indian Board for Wildlife 1952, Wildlife Protection Act, and others I have listed. What is the most important? Environment Protection and Biodiversity Conservation Act 1999. It covers 3 aspects: EIA, Environment Impact Assessment, biodiversity protection, and protected areas conservation. 3 aspects. Now, how to achieve the goal of biodiversity conservation? I have listed here 4: implementation of proper protection measure to facilitate natural regeneration in the forest, because I am botanist. Now, launching environmental awareness program for rural community to educate them. Then integration of the formal science with the informal, means involvement of the scientists, NGOs, and local indigenous community to work together. That will be very much effective. Now indigenous people be rewarded and credited for their unique knowledge on the traditional medicines. Because in some elderly aged people in northeast tribal states, this medicinal or herbal plant knowledge is inherent. But we are not giving due importance. If we are documenting the plants, so we are not recognizing them. So this is our duty and government duty to recognize them so that the interest can continue in the younger generation, because the young generation is inactive in this regard. So then actually this knowledge is on the verge of extinction. Now, sustainable development. Madam was talking, just I have given one figure. This is the meeting the need of present generation without compromising the need of future generation. And earlier I told Professor Yadav actually mentioned that how last. And I am sir within well within the time.
That is why.
Yeah. And this sustainable development, the most important effort is the sustainable utilization. If you are, we are utilizing the biological resources in the sustainable manner, then the sustainable development and conservation of biodiversity or environment or anything is very easy. Here, Madam talk about social, economic and environmental aspects. These three pillars are there for the sustainable development. And the environment acting with the social, that is only bearable. Social with the economic, that is only equitable. Environment with economic, that is only viable. When these three forces are acting together, that is only sustainable. And last slide is the significance of biodiversity. This is the theme of the Science Congress. I have listed here to preserve genetic diversity, to ensure sustainable utilization of genetic resources, to ensure community stability and productivity, to maintain ecological process, to preserve endemic and threatened species, to restore degraded ecosystem, to maintain groundwater table, and last, very important, to ensure cleaner, greener and eco-friendly environment. With these lines, I complete my presentation, sir. And I am very thankful to Professor S.K. Chaturvedi Sahab for calling me and facilitating also to meet actually my faculty colleagues from different organizations. Thank you.
Please, sir. Thank you very much, Professor Mishra.
Yeah, they are in UNESCO World Heritage Site.
Yeah, near Cherrapunji side. They have studied.
Not studied.
They have not studied.
Come, you start. Aryaji.
Now I request Professor Avinash Ane sir to felicitate Professor B.P. Mishra sir.
Sir. Yes, please. Yes.
Thank you, sir.
Thank you. Now, last presenter is Professor N. Mathivanan. He is Director and Head Center for Advanced Studies, University of Madras. Formerly, he was Registrar, University of Madras. Now, Professor Mathivanan has 28 PhD students in his credit. He produced 34 MPhil students. He has published 110 research papers in very high impact factors. His H index is 33 and citation 3912. He had 22 projects and he generated nearly 7 crore of rupees out of his projects. He is a fellow of AMBO Japan Fellowship, INSA, DFG Fellowship, DAD Harmony Fellowship, then DBT Associateship. He got Outstanding Scientist Award on Noni, Tamil Award Tamil Nadu Scientist Award in 2016. He got Professor S.N. Banerjee Memorial Award by Indian Mycological Society. He is Fellow of National Academy of Agricultural Sciences. He is Fellow of National Academy of Biological Sciences, Fellow of the Academy of Sciences, Tamil Nadu, and he is Fellow of Royal Society of Biology, UK. Now I request Professor Mathivanan, please come and speak. The topic of his deliberation will be harnessing potential of medicinal plants for human health. Thank you, sir. I request you to complete within 20 minutes.
Good evening to each and every one of you. Thank you, Chairman, for your introduction as well as permitting me.
Actually, he is a special invitee by ISKA.
Yeah, I have been invited by the President of ISKA as well as the organizing secretaries of this great university to deliver a special lecture. But unfortunately, my abstract, though I have sent it my abstract, biodata and photo, but that has not reached to the President. But yesterday I met him and he kind enough to accommodate today. Therefore, I am here and I am extremely happy. I am the last speaker. It's very difficult to make you to sit and listen. But anyway, for the students, at the end I would like to give some important message. So therefore, if you are here, then you will take the message. Otherwise, if you want to go, you can also go also, because now the time is for cultural program. Anyway, I will be sticking on my time, 20 minutes. This is the conference, 108th conference, in which I would like to present 3 slides before going to my actual topic. The first slide is Center for Advanced Studies in Botany in the University of Madras. This is a 165 years old university, one of the pioneering university in the country. In the year 1857, when Britishers were there, this university has been started, along with the University of Kolkata and the University of Mumbai in the same month, September. My university started September 5th. And this university has produced 2 Nobel laureates, you know pretty well, Sir C.V. Raman as well as Dr. Chandrasekhar. Both have been given a Nobel Prize in the field of physics. Both of them are alumni of University of Madras. Not only that, this university also produced 5 past presidents of our country. 5 Rashtrapatis of our country as produced by University of Madras, 2 Supreme Court judges, and a large number of professors, scientists, administrators, etc., etc. So, University of Madras, we have Center for Advanced Studies in Botany. It has been started in the year 1930 as a university botany laboratory, as a research laboratory, not a teaching department. Later only we have included teaching, but initially it has been started in the year 1930. Now our department is having, 92 years old. Under the Center for Advanced Studies in Botany, why I am presenting this slide? Because this is relevant to the core theme of women empowerment. We have produced 552 PhDs so far. Out of 552 PhDs, 196, almost 35% are women. That is the point I wanted to inform you. And not only that, 296 MPhil we already produced. Out of 296, 182, that is 61.5% are women. So, women empowerment really we are doing. Another slide I would like to present before you, a great botanist, a celebrity scientist. You all very well known about Dr. E.K. Janigammal and her contribution. I am also today came to know that Dr. Professor Yadav sir has received the prestigious E.K. Janigammal award. But you know pretty well about her contribution in the field of cytology, plant breeding, biodiversity, and also ethnobotany. But one thing I think most of you may not aware, her last stage of life, she spent at our botany field research laboratory, University of Madras. This photo, I think, this photo has been taken in our botany field research laboratory, where she has been awarded a DST project on ethnobotany. She was handling that project, and she died in the botany field research laboratory. The University of Madras Professor Mahadevan, then director, has given her accommodation as well as the laboratory facility. So therefore, we have a very close collection with the E.K. Janigammal. Not only that, she is the alumnus of University of Madras. She graduated from Queen Mary's College, Madras, and also worked at women Christian colleges at Madras for a short while, which are affiliated to University of Madras. So, and I am happy. Yesterday, I took this photograph in the stall. Actually, they put this portrait here, and also with the sugar pot, where she has done a lot of work on sugarcane breeding. So I am extremely happy. And coming to my actual topic, harnessing the potential of medicinal plants for human health. I am not a medicinal plant scientist initially. I am a plant pathologist by research. But later I compelled to start the work on medicinal plant because of my botany teacher. Undergraduate teacher, he has told about one plant. Therefore, I started during my research, as well as University of Madras has been given a black sheep program called University with the Potential for Excellence. You know pretty well about that project, very big project. We completed 2 phases of that project. That project mainly on the herbal plants, development of drugs from medicinal plants. I was the chief coordinator for the second phase, and also centered with potential for excellence in the particular area that has been given for life sciences department. 4 department, we jointly got that project. I was also coordinator for that. That is also mainly on herbal plants. Currently, we are running on RUSA 2.0. 1.0 was completed, 2.0. We got about 50 crore rupees under this program. I am the overall chief coordinator of this program. Again, we are working on herbal plants. So, therefore, I would like to present some of our research. Prior to that, I wanted to highlight. This is my outline of my talk. Importance of plants, importance of herbal medicines, phytocompounds. Before going to my actual research outcome, I would like to give some information about the potential drugs which have been already isolated and identified from the plants, and also conclusion way forward. Coming to the importance of plant, you know pretty well about the importance food, fiber, and wood. These are the primary needs of a human. Along with that, and plants are also responsible for production of flavors, fragrances, pigments, etc., etc. There are so many. Only some products I am listed here, and many more also, in addition to ecological, etc., etc. benefits. Coming to this slide, I wanted to emphasize. Actually, we eat grains, vegetable, fruits for nutritional purpose. But the plants not only serve as a nutrition, and they are also serve as a health benefits, because these plant produces contain various phytocompounds along with the primary metabolites like carbohydrate, protein, and fat. There are different phytocompounds. So those phytocompounds are having health benefits. And therefore, if you consume food, and we will also have health benefits. So therefore, food is medicine. And I purposely put this slide because mostly a doctor used to say, don't eat too much of rice because the glycemic index will go high in your blood because it contains starch. But not every time is right, and rice is also having various phytocompounds, and some rices are good for even providing health benefits. So therefore, every plants are good for us. Coming to the herbal medicine, you all very well aware, it is very important component in the healthcare. Almost 80% of the global population, its literature says, directly depending on plants for everyday health needs. And even 30% of the doctor prescribed medicine, even in the developed countries, contain phytocomponents. So I will give you some information. Coming to herbal medicine, we can classify into traditional medicine. We have traditional systems of medicine. We know we have Ayurveda, Siddha, Unani, etc. And also local community. They also have their own knowledge, including grandma, the Vaidya. So the grandma sitting at home, she used to have her own preparation. That is also comes under traditional medicine. And coming to the modern drugs, through organized R&D, research and development, we will identify the bioactive molecule, and it will go different stages of evaluation, finally clinical trials, and coming to the market as a drug. Coming to this way, how to produce this bioactive molecule through R&D research. You can take the crude extract, probably different solvent we can use to take the extract, and we can purify the compound, and we can do the chemical analysis and identify the molecular structure, molecular weight, etc., etc. And each and every stage we have to go for in vitro study. There are assays are available for each every the issues. And once if you prove the in vitro it is acting, and it is also undergo in vivo study using animal models, and that is not sufficient. Both in vitro, in vivo is not sufficient. We need to do toxicology, acute toxicology or chronic toxicology we have to do. Then also we have to elucidate the mechanism, how this bioactive compound is acting. What is the mechanism? That has to be elucidated, and also bioavailability. If you give this the bioactive compound, how long it will stay in the body, then whether it is available for the treatment, whether it is creating any kind of residual toxicity, those things have been elucidated, and then it will go for clinical studies. There are phase 1, phase 2, phase 3, phase 4, something like that. We need to do clinical study. Clinical study alone may not sufficient, then we need to go for commercial feasibility, whether this compound, when we put it in the market, whether it will give you profit. So that has to be done. So once everything is over, if we confirm this molecule is going to give profit, then it will be released as a drug. So it takes long time, maybe 5 years to 10 years. That is so long time it will take. Coming to the pharmaceutical potential of plants as a whole, there are 391,000 plants according to the Kew Botanical Garden have been discovered so far, but not all the plants have been scientifically evaluated. Out of these many plants, only 17,000 plants we are using for our health purpose, and among these 2,500 plants screened pharmaceutically, we already identified more than 1 lakh bioactive compound. Not all the bioactive compound qualified as a drug. Only few have qualified as a drug, but we have identified them. Some of the drug, the bioactive compound I am presenting before you. One such molecule is curcumin. You know pretty well. Turmeric is our important medicinal plants. It contain curcumin or curcumin, which is having various bioactivities. Similarly, there are many more molecules have been discovered already. I would like to present some of the molecules have been already commercialized as a proven drug. For example, this quinine has been isolated from Cinchona officinalis bark from 2 French scientists in the year 1820. But this is the person, Samuel Hahnemann, who is the father of homeopathy. He has tested its anti-malarial activity. Even today it is available as an anti-malarial activity alone or its derivatives. Coming to the other molecules, aspirin. Aspirin, everybody knows about its analgesic effect, pain reliever, etc. And also it is good for heart patient. Even doctor used to advise you keep aspirin every time. When you have any kind of symptom, you put aspirin, so go to the hospital quickly. This has been identified from a meadow sweet. It is a common weed plant by Felix Hoffman in the year 1897. Another interesting compound, you know pretty well, Catharanthus roseus or Vinca rosea. Previously, wind blast and increastin. These 2 compounds have been discovered by Robert Nobel from the Toronto University, Canada. And this is a proven anti-cancer drug. Even today it is available for the treatment. Another important compound is camptothecin from Camptothecin acuminata. From the bark they isolated Monruval and Mansukh Wani. Very interestingly, Mansukh Wani graduated from University of Bombay. And he has moved to US and settled over there. He is a chemist person settled over there. Along with this person, they have discovered this camptothecin anti-cancer molecule. Another interesting point here is same Mansukh Wani and Monruval joined with Arthur Barclay, they discovered this Baclitaxel from the Taxus brevifolia. This is a blockbuster molecule. Left and right people are selling this molecule for the cancer treatment. Very, very interesting outcome from the bark. Another wonderful molecule, metformin. The diabetic patient without a metformin, their day will not end. I am damn sure. Every doctor is prescribing metformin by the diabetic. This has been discovered by a physician, French physician Jean Streen, from the Linga officinalis. Actually, this is from ethnic knowledge. Actually, he found out some tribals are there taking the root and taking decoction and drinking, and he found out why. And then finally, he came out with the metformin. It has now sold extensively globally, in India also. Coming to another compound is Artemisia annua, from Artemisia annua, artemisinin. This has been discovered by Tu Youyou, the Chinese scientist, for the control of malaria, for which she has been awarded Nobel Prize in the year 2015 for medicine. So, such a wonderful things I presented before you. Therefore, why I am telling here is, there is a potential in plants, medicinal plants. They are actually serve as resources for the health benefit. Actually, what we have done at our biocontrol and molecular metabolites lab, because I'm basically a plant pathologist, and I compelled to start this research, and we have done biodiversity, antimicrobial activity, antioxidant, wound healing, hepatoprotective, cytotoxicity, anti-snake venom activity, anti-candida, anti-tuberculosis. Some of the results, I'm not going to present everything because it takes time. Just glimpses I wanted to present before you. I start with this one, Morinda pubescens. This has been introduced by my BSc professor, and he only told about this plant. It is a very common plant. Everywhere you can find out. But it has a lot of medicinal property, but nobody is aware of it. They have not done any research. We have done quite a few research. And one such research is hepatoprotective. We have taken all these the biochemical parameters, and we found out, and hepatoprotective effect also. Then we have published this in medicinal chemistry research. Another interesting results I wanted to present before you about the wound healing activity of Morinda pubescens. You can see here. If I show the picture, you will appreciate. You can see here, this is the control wound excised rat. See here, this is the control, and this is the 10 mg per ml crude extract of fruit, and also 20 mg per ml crude extract, the fruit extract, and we have treated. After 15 days, still the wound is there. Whereas 10 mg or 20 mg, if you treat, within 15 days the wound has healed, and even the new hair has formed. That's a wonderful activity. So that is the one we have. This also we have published already. Coming to the another plant, noni. So for which I have been given Outstanding Scientist Award, because I have started noni research for the first time in the country, along with my friend Dr. G.R. Singh, who was also Vice Chancellor for Chaudhary Saran Singh, some university in UP. Now he has relinquished his office. He is from Andaman. He has started, and for which we have done some lot of work we have done. But I wanted to, actually very interesting, I wanted to present. Noni is the Polynesian name, but the English name is Indian mulberry. Actually, this is, people are believing that it's an Indian origin. Some people say it is originated from Australia, Darwin area, but people from USA, they believed, and it is originated from India. They mentioned one professor from John Hopkins Hospital and University came to India by name Neil Salman to see this plant. And he has mentioned in his book, I read that book, he has mentioned it is originated from India. Anyway, we are not aware of this plant till 2003. And 2003 we have started, and you know, it for the past 2000 years, the Polynesian people are using this for various health benefits. In fact, I personally visited Hawaii and Polynesian countries, and I have seen how they are using it. I will show you some photograph. You will appreciate. There are many products are available in the market. Nothing is known, and it is already available. And you can see there are many more products, different types of product. This photo I have taken in the Papiti, the capital of Tahiti, the small island. It takes 5 hours from Hawaii to reach that particular small town, and where they are producing and selling. These are the Polynesian people, and even we purchase some more product. There are soaps, so many products they are selling in the market. We purchased from. This is a factory. Actually, this is the first company, Tahitian Noni juice company. First time they have commercialized this product from the Tahiti. But the company is in USA, but they have a very big factory. This is a back factory. They produced the noni juice, and they export to USA. From there they manufacture. 240 plus products are available on noni. I don't know how many of you know. And these are some of the Indian products are there. I am not propagating, just I am bringing to your knowledge. These are the products are available. Coming to research, what we have done, Noni, in collaboration with we have done many work, and I wanted to highlight only one aspect, that is the anti-proliferatic activity of Morinda certifolia on retinoblastoma cell line. And this research we collaborated with the Department of Ocular Pathology and Cell Biology Laboratory. Vision Research Foundation is a part of Shankara Nethralaya. This is the premier eye hospital in Chennai. Many people from even north, they go to Shankara Nethralaya for the eye problem, and we collaborated. They have a Vision Research Foundation. One of my students, Dr. Surendra, now he is in Canada, working with the St. John's Hospital at Newfoundland, and he has done the entire research in Shankara Nethralaya itself. They have excellent facility to work on it. This is the retinoblastoma eye cancer, very, very, very serious problem in children up to age of 5. It's very, very pathetic situation. If I show the next slide, you will be scared about it. See this, so much of tumor, very big tumor. I am not purpose is to scare you. I wanted to highlight the amount of panic the children used to undergo. Not only that, it is invasive. Even both eye will affect, and finally the children will die. So far there is no treatment. There is no radiotherapy. There is no chemotherapy. There is no surgery. That is the beauty of this, the problem of this. And when we approach Shankara Nethralaya to do work, they have a foundation. I have presented 3 times. They don't want to take up research on the medicinal plants. So finally we convinced, and finally we take up this research, and we did the work in vitro condition, and we did the crude fruit extract, and also tested in the cancer cell line and normal cell line. You can see cancer cell line, normal cell line not affected, cancer cell line have been inhibited. All the cytotoxicity have been established, membrane blurring or shrinkage of cell, granular formation, these and all we have seen. And even we have confirmed using annexin style the cell death very clearly. And then we wanted to isolate the compound. What is the compound responsible for it? And we did it, the column chromatography purification technique. It takes a longer time to purify. And finally we got a compound. Purified compound, and we tested this compound. This compound is chemically we characterized using all the spectral data. Finally, we end up with the scopolamine. Scopolamine is not a new compound. This has been already reported in many plants. But for the first time, we established this anti-cancer, cytotoxin. Now, I am not saying it anti-cancer activity because it's a big name. We need to do all this animal study, but we have done only the cell line study. You can see here, again, the purified compound as well as the crude compound is effectively inhibiting the cancer cell line, but not the normal cell line. And we also went further to do the DNA microarray analysis using Iceland platform. There are lengthy procedure we have to follow. This is the hybridized slide, and we found out the analysis that the data shows these many genes were downregulated and these many genes are upregulated. It shows that many of the cancer inducing genes are upregulated. The cancer suppressing, cancer inducing genes are downregulated, cancer suppressing genes what we have are upregulated. So both way it is helping, and this data, the whole genome data, we already submitted to the NCBI website. And finally we found out the scopolamine as well as crude extract was very, very effective in inhibiting the growth. Unfortunately, we don't have an animal model because this is very specific for human. So therefore we could not progress further. We left out with nothing except showing these results. Coming to the another research, very interesting research is anti-synaquinum activity. And this has been done in collaboration with the Konkan University, Thailand, and also the King Institute of Preventive Medicine and Research, Chennai, because this institute is responsible for production of polyvalent antibody for the snake bite. So they use a horse to produce it, and also supported by Gencor Pacific Organic India Private Limited. This is a company, US-based company, but their office in India, and they are supported with 40 lakhs rupees, and also the ELISA reader they are given as a gift. The research has been done by Dr. Armu. Now he is a director of the company, and he has done this entire research. And what is this number? 2,50,000 is a 2008 to 2004 data, but now this number has been increased to 58,000 last year. 2 lakh people are bitten by snakes every year, and 58,000 people dying every year in India. Nobody knows about it. That is a pathetic situation. No policy, nothing. Nobody is interested because why? It is happening in the rural areas, agricultural fields. So therefore it is considered as a tropical neglected disease as WHO. WHO has been coined this disease as a neglected tropical disease. Snake envenomation as disease of poor people. If it is rich people, people would have put money to do research. In a developing countries, it is categorized as a neglected tropical disease. So therefore, no awareness, nothing is there. Even when Arumugam came to me, his ethnobotanist came to me for doing research, I said, no, nothing doing, because I am a plant pathologist. I don't know anything about anti-venom activity. But he was very particular because he was working in a different institution in Kerala, like different Kottakkal Vaidhyasala, etc., etc. He gathered so much of information. So therefore, he was very particular to do this research, and these are the big four snakes, very, very poisonous snakes we have. And the available treatment is the polyvalent antibody. Otherwise, they will go to only the local healers. Many of the patients, almost 90% of the snake bite victims have been treated by the local healers with the medicinal herbal treatment. Still, a large number of indigenous medicinal plants used by the tribes and traditional healers for snake bite treatment are yet to be scientifically evaluated. They are doing it, but scientific proof is not there. So therefore, after seeing this information, I was convinced to do this work. And he is the owner, I am just a facilitator. And then the journey has started. This is 10 years of research. It's not a small thing. I will be presenting in one slide. We have collected 76 indigenous medicinal the plants based on the local knowledge. So we have surveyed. We got the information from the local people and tribals, and also we made a 92 methanolic extract with the different parts of the plants, and then we don't know what to do further because there is no assays available, not very clear assays available. Then I told Arumugam, look, you do some antimicrobial activity, finish your PhD, and because there is no methodology to do this research. Then by the time we got a paper, a ray of hope came in the form of paper published in the year 2005 in Biochemistry Journal, by the Konkan University, Thailand Biochemistry Department in collaboration with the Natural Research Institute, UK. So that paper says that the ELISA screening can be done for anti-snake venom activity. So then I rang up to Professor Nissan Satyasai, wonderful professor, Nissan Satyasai, and his wife also professor in pharmacology. She is Jindana Satyasai. Both they agreed, accepted. Then myself, my student went over there, and we did ELISA screening in his laboratory, and we shortlisted top 10 plants, and then another trip, Arumugam alone went and he did a non-dissociative page electrophoresis. It is the first time we have performed this technique. He has developed in his laboratory. You can see here, this is a very big slide, but this is the venom alone. You can see venom contains different proteins, and if you treat with the different concentration of our plant extract, see here in the higher concentration, the venom protein has been denatured. So that is the beauty of this one. Then this is the in vitro study. This is also in vitro study. Then we taken this into ex vivo model. So we have to excise the rat pheneric hemidiaphragm, and also then we subjected to the machine, and it will give you a polygraph. So based on the polygraph, we can say whether it is having activity. That has been confirmed by another test called SICK Bivendor. And for your information, only 3 institutions in the world having this facility. One of the institutions is Konkan University, and they are doing extensive work on anti-snake venom activity. Yes, yeah, finish it. So then we also went to the King Institute of Preventive Medicine, where polyvinyl antibody is being produced, and we did animal model extensively, and we confirmed. And this Talbergia corrida bark extract is having very good activity. And for your information, this information we got it from the Kani tribes in Tirunelveli district, and what they do, before going to the forest, they take the bark and chew it, and they will go. They chew. And they believe even snake bite, no poison will kill them. So that is the information, and this project is wholly funded by the industry, and then we made a MOU with them. Recently NBA also got approval for the patent. We applied the patent, we got a global patent. Many countries they already given the patent, and very interestingly, this company, if we are going to commercialize, and 30% of the royalty going to give to University of Madras, and out of this we are going to give 10% to the Kani tribes for their development. That is the arrangement we already made. That is very, very heartening for me, because this research out of 28 PhD students, this is the one I really valued very high. And then quickly I finish it, sir. This anti-candida, this is also very interesting, very important. Every part of a body will be affected by candida from top to bottom. And not only that, even the valve and the catheter, whatever the implant also colonized. And this is the second most infectious problem for the death. And we have screened the same plant extract, whatever we have taken for anti-cyanic venom activity. We screened and also we purified 2 compounds. And interestingly, you can see the crude also showing activity and purified compound. This is the one crude extract, then fraction number 9, compound 1 and compound 2. 2 compound we purified. You see here amphotericin B is a multi-drug resistant pathogens. Candida tropicalis, Candida albicans. No drug is able to inhibit. You can see here very clearly it's not inhibiting. So clearly, even we tested against the clinical trials, we got 7 clinical isolate from the Kilpack Medical College. This is the summary. 180 plants we screened, and finally we end up with a 404 plant. I am not presenting the name of the plant because this now we given all the data to the attorney, patent attorney. They are writing patent. We are going to file a patent. Why? Because we got 2 new compound. No, this compound has not been reported so far. And thing is, we also elicited the mechanism. The ergocerol biosynthesis inhibition and also the Eiffel formation, yeast to Eiffel formation. If it's yeast, it's not pathogen. It's Eiffel, then it is a pathogen. That also we elicited a biofilm formation, and also it's not toxic. And that also we proved, since it's a novel compound, we are also in the process of patenting. So therefore, I am not presenting. Maybe after filing patent, I will present it the next time. Very And coming to the anti-tuberculosis activity, only one slide. You can see this also another interesting research. And this is the plant is not available in India. We got this crude extract. One of my friend from abroad, he has given as a donation for screening for some other purpose. But while screening, we found out it is acting against Mycobacterium tuberculosis. Interestingly, multi-drug resistant as well as extremely drug resistant Mycobacterium tuberculosis. Therefore, we have collaborated with the National Institute for Tuberculosis Research. It's in Chennai. It's a national institute under ICMR. Recently, this Mrs. Priyadarshini, she has submitted last week her PhD thesis, and this also we are now going for patent because we purified compound, we elicited mechanism and all. For your information, even the NIRT people are very surprised to see the activity. And more information, then some 6 months back, Hindu, there was information about the distribution of the tuberculosis in the world, in which I have seen, particular the geographical area, TB was not reported. And exactly from there, we got that material. That shows, because the people there, they are taking as a salad every day. There is no TB reported. So there is a direct relation in the medicinal plants. So therefore, there is a lot of potential. Finally, I actually mentioned 391,000 plants have been reported as per Q. Only 17,000 plants we are using as a medicinal plants. So therefore, there is a lot of potential. Young botanists are urging, see, you can take this plant. This is also unnoticed plant, 2003 before, but there is a lot of products are available out of this. Another particular plant is Sibaktan. Even Dr. Ramesh Dandan has mentioned, he was working on this. This plant also unnoticed, underutilized plant some years ago, but now they are making a product out of this fruit extract. It contains omega 3, 4, 5, 6, 7 fatty acids. Only we get from the sea, the fish oil, but here it is there, vegetarian omega fatty acid. Last one, sir. Yeah. This is the one, blueberry. Nobody, actually it is available now, every supermarket, but 1900 before nobody was knowing about it. It's only ornamental plant in USA. But now it has been extensively commercialized for various health benefits. There are many. And finally, the editorial Nature plant, they mentioned, they written in the year 2020, February, after this COVID pandemic, what they mentioned. This is one page, but I have taken only abstract. Plants are important not only for food, but also for medicine. Understanding the taxonomy, sir. You can happy. Taxonomy, ecology and conservation of herbs, as well as the pathway of secondary metabolic synthesis is important for drug development. Investing in research into ethnobotany, phytochemistry, plant physiology and ecology will be vital in protecting the global population from current and future pandemic. That is the editorial note. Future pandemic also we can manage using the medicinal plants. So that is the message for the youngster I wanted to show. Finally, medicinal plants are always pivotal source for human health, and discovery of bioactive compound from medicinal plant need to be done on priority, which eventually lead to provide health benefits. With this, I thank profusely the organizers of this 108th Indian Science Congress and the president and also chairman, and also for you for your passion. These are the people. These are the collaborators. These are the owners of the work. These are my collaborators. And finally, I thank the funding agency and thank you everyone. Thank you.
Okay, sir. Thank you very much.
Now I request Professor R.P. Thakre sir to felicitate Professor N. Mathivanan sir.
Thank you.
One more, sir.
Important announcement for the delegates that we have a small water body which is called as Futara Lake, and recently honorable was 6 p.m. Tomorrow also the same timing would be there. If you can show your delegate or speaker card, and that will be your entry pass for watching the fountain show. आज होता आणि उद्या इसे होगा.